I was unmuted online. Good morning to the virtual audience as well. Welcome to the first ever Nextflow Symposium happening on the Wellcome Genome Campus. It's wonderful to have you all. A couple of things before we get started. Because we care about your safety. So there is a defibrillator and a first aid kit just outside the lecture hall. And there's a water fountain beside the reception desk. In case of fire, please follow the green signs which say fire exit, and wherever you are in the campus, you would find them. And there is an assembly point outside the main reception hall. And if you have stated any dietary requirements, please note that your food may be plated separately. Check for the labels. And if anything has changed, please report to the reception or any of the committee members. And to all the presenters, I humbly request you to upload your presentations if you haven't done that already, and fill the model release form. For the audience who are not on campus, who are visiting the campus, there is WGC guest that you can register to. This is the Wi-Fi that you can register to. Now, without further ado, let's get started. So today, firstly, we have opening remarks by Andrew Yates. Andrew, fondly known as Andy, leads the genomics technology infrastructure team, which provides technical development and production capacity for the Ensemble and Decipher projects. Andy joined Embel EBI in 2006 and has developed production infrastructure and databases for Integrate, Ensemble, and Ensemble Genomes, which he was a founding member of the resource in 2008. He became a team leader in 2015 and now serves as joint PI for the Ensemble project. Since 2017, Andy has led the Global Alliance for Genomics and Health workstream, Genomic Knowledge Standards, and has steered the development of 3 standards: RefCAD, Variant Representation Specification, and Service Info. His background is in pharmacology, and he holds a master's in science in bioinformatics from the University of Manchester. Please welcome Andy. Over to you, Andy.
Thank you so much for that lovely introduction and making me blush so much. And welcome to everyone to the first Nextflow Symposium, especially to those who this is your first time visiting the campus. Please, at times, take advantage, have a look around. You have some amazing architecture, some amazing buildings, the conference center at the far end. The roof over there is actually based on the roof in the British Library by the same people who built it, I think, not by the same architects. It's a bit like getting in someone that says, oh yeah, you know, I built the Sistine Chapel. Cool. All right, well, I'll get the guys that built that then, shall I, rather than ones that designed it. But it is beautiful over there. Please, please, please go over there. And to everyone online, thank you for joining as well. It's fantastic to have you all here. Okay. There's a big thank you to our supporting organizations, the Wellcome Sanger Institute, which is conveniently located over there, EMBL EBI, which is conveniently located right here and over there as well, the Wellcome Genome Campus, in which we're all embodied within, and also Nextflow. Without the support of these organizations, we basically would not be having this today. And to those who have helped organize the symposium, all these very lovely people, especially I have to say a big thank you to Priyanka as well, who is basically the driving force behind organizing this and has been bugging me like mad for trying to organize things left, right, and center. So thank you so much. But also thank you to the other organizing members as well. Again, without those people, this would not be happening today. And to our speakers, I'm pretty sure there are Oh, I'm pretty sure there's some pictures missing here. If a speaker's picture is missing, it's because you've done a very good job of scrubbing your virtual presence from the internet. So congratulations. But you'll be hearing from all of these lovely people today. Okay, very quick quiz. So show of hands in the room. How many participants do we have both in person and online today? So if you think it is 50 people, just quickly raise your hand. Nope. 75 people? Ooh. 100 people? 125? Well done to the 125. 125 people. It's absolutely amazing. And how many organizations are participating today? Knowing full well now there's 125 people actually participating. Is it 16? Show of hands. Cool. 22? 34? 47? More. One soul more in the room. It was 47. Those of you who know me know that I like setting good quiz rounds. Well, okay, not good quiz rounds, but I like setting quiz rounds. So yeah, we really have a diverse set of participants from across the world, from very different nations as well, from both academia and from industry as well. This is a very diverse group of individuals. I think there's a lot to learn from everyone today. So briefly about ourselves, because being up here, I want to take advantage of that. So what is EMBL EBI? We are one of the world leading repositories of biological data and biomolecular data, and we are part of the European Molecular Biology Laboratory. We're one of 6 sites, and all of these are based around Europe, from Germany, France, Italy, Spain, and of course here in Hingston. Our mission, well, we are delivering data resources. We want to perform research. We're training next generation scientists, engaging with industry, and coordinating bioinformatics across Europe. And of course, events like this are really supporting the pillars around training next generation scientists, engaging with industry, but also actually helping to deliver our data resources as well. So really, and actually even down to the research as well, because we are completely computational here at EBI. So Nextflow, workflow managers play an important role in our lives. And of course, because this is a bioinformatics community, probably many of these resources you use daily, perhaps if not daily, I am sure weekly, I hope so. But we cover such a broad range of different resources. Now, in terms of the major challenges that exist out there, I just want to take a step away to think about some of the areas where we are trying to challenge some of the big issues in the world. So precision medicine, human health, trying to improve patient outcomes. That goes through the work of our research groups, but then also through the provision of resources like Ensemble and Decipher. Pathogen impacts, which is helped through our pathogen portals, but then also moving towards the future in terms of AI and high-level curation of our data resources and providing that for AI learning toolkits. Now, the fact is, though, that without good workflow managers, without good ways of executing high-volume compute, we could not do any of this. Now, I'm not originally a Perl programmer, but many years in the Ensemble project taught me to be a Perl programmer. And the guy who wrote Perl, Larry Wall, once talked about the 3 virtues of a great programmer. Now, this is a slight tangent, but it will make sense a little bit. So the first one he said is that great programmers are lazy. And that's the virtue that basically means that you want to script, you want to program, because doing it manually is just boring and tedious, and it will go wrong. Impatience. This is when you're basically just getting angry about your computer. It's like, I just want programs that don't react to my needs, but actually anticipate them, or at least they can pretend to anticipate what I want them to do. And finally, hubris as well. I want to be proud of my code. I want to be proud of the work that I'm doing. I don't want anyone to say bad things about my code. So 3 virtues of good pipeline software. They should have ways of allowing me to reuse that code. I should be able to share. I should be able to distribute that. Impatience. I should be able to get my jobs running somewhere. I should be able to burst. I should be able to do the processing I need to do because I'm lazy and I need it done now. Oh, sorry, I'm impatient and I need it done now. And hubris, I want to write good quality pipelines just in case the local chainsaw wielding maniac bioinformatician decides to drop by. This is one of my friends who always said, programmers, if you believe there is a chainsaw wielding maniac somewhere nearby who actually will kill you if you're trying to do bad code. So, okay. But those three things are important. And Nextflow and Nextflow Core are actually helping us to achieve those. The ability to share those pipelines, the ability to actually allow others to replicate or reproduce the results that we're producing. The fact is that there's an active community there, that training's provided and plentiful, and there is support available as well. Now, the other thing that this symposium has come out of is the Wellcome Campus Pipelines community. This is started in around about mid-2023. There are three kinds of events that have been held. There are coffee drop-ins, there are use case seminars, and then there is this event. The lovely people on the right-hand side are all organizing members of that community. If you're not part of the community, then I really encourage you to go and talk to them as soon as possible, because they've got little badges. I didn't know what the badges would look like, so this is my best guess on what the badge might be. I can see that it's a bit less lanyardy in the end. Oh, well, never mind. But please keep an eye out for these people, have a chat, join up. Sharing knowledge is important. Okay, and finally, because I can see that we need to be moving on here, what I hope you'll get out today. I hope that you're going to get some insight into some of the biggest challenges that are facing our industry today through the people and the diverse participants that we have talking. I hope you get a chance to talk to people you've never talked to before. I hope actually you get a chance to talk to people that you haven't met before and quite like as well. But I really hope that you just spend that period of time talking to someone that you haven't talked to previously. Also, a few tips and tricks around Nextflow and some ideas that you might go, oh, that's a nice idea, that is. And finally, I want you to find ways of being lazier and impatient and embody hubris as much as possible, but not to the point of being dangerously overly confident, as I feel like I am at the moment. And with that, thank you so much. And I will pass over to our keynote speaker. You're gonna do your, yeah, sure.
Let me share my screen.
Sure. Yeah,
Yeah.
we will stop the screen here.
Okay, I will make. Wait, is it the? This one.
It is my great pleasure to introduce Paulo, who is going to be our keynote speaker. Paulo is the CTO and co-founder of Sequera Labs. He's a computer scientist with a strong interest in high-throughput scientific computing, data-intensive applications, parallel programming, cloud computing, and containerization technologies. He has broad experience as a software engineer and software architect in life science and healthcare applications. He's an open source advocate and the creator and maintainer of the Nextflow workflow system. Over to you, Paulo.
Hello. Thank you for organizing this event. I'm very excited to participate here, and above all, also thank you for inviting me to present this work. Really a big honor to talk about Nextflow here at Shanghai ABI. And also it's quite important event for this project, because we are celebrating 10 years since the first commit on Nextflow as an open source project. Project was started maybe in 2012, but was made open source 2013. So it's a good opportunity to say thank you to everybody involved into this community, everybody that commit code Nextflow, provide feedback, or just use every day like you are doing here. And so this is a very big opportunities again to say thank you, all of you. And also what I like to think sometimes about Nextflow, how this project managed to grow from a small idea originated into a small research lab Barcelona into something much bigger. And that is all. And then the main idea behind this project was to allow the scalability of data pipelines, removing all the friction that there is behind enabling the data pipeline. So, but also above all, allowing scientists to focus on their work to allow collaboration. So these are the main idea behind this framework and what we continue to work every day, that we continue to develop this technology. The point is that we are going forward, or we are going toward more complicated, more complex deployment scenario, which you want to use more cloud, you want to write more complex pipeline, you want to scale more your pipeline. So this is why we are evolving this technology from a single tool or single workflow engine to something more complex, to an ecosystem of technology in which there are different components, in which these components play a role to optimize the execution of the pipeline that you need to run and to deploy. So I'm going to talk today about a bit more about this technology, of course, about Nextflow, but also about Fusion and Wave, that are two new components that we are adding to this stack of technology. And the idea to start with this Fusion file system, and why we need a special file system for Nextflow data pipelines. Because the real challenge for the deployment of scalable pipeline in the cloud, but not only in the cloud, is the storage. It's not much the problem of computing. Computing is pretty easy to have. You can have just credit card by a lot of cloud computing resources, so it's not the limiting factor. But the point is how we can use computing in an efficient manner. To have efficient computing, we need an efficient way to manage data transfer into the deployment of our pipelines. Because at the end, these data pipelines ingest a lot of data, produce a lot of data, so it's key to have an efficient way to move data in an efficient manner, optimal manner. So there are a lot of shard file systems, distributed file systems in the cloud, but most of the time they were designed for a different work. They were more designed for HPC. So they tend to be expensive. There are other services to be maintained. This is something that we would like not to have. Instead, the ideal solution when we deploy a pipeline in the cloud is object storage, because object storage, like S3, is designed to scale, is relatively cheap. But the point is that there is a mismatch between the interface used by object storage and the interface of the tool that we use in our pipeline. They expect to use access data just using local files. So how to solve this problem? And here where comes this Fusion solution. Basically, essentially, is a kind of adapter that make it possible to access data over an object storage like S3 bucket or cloud storage bucket, like if it were a log file system. So when you have S3 bucket some files, you can access locally using a virtual file path, the /fusion/S3 and the bucket name. And then behind the scene, there is this driver that automatically transforms all the access to this file path to IO operation in a native manner using the API of the underlying object storage. But at the same time, also, this removes all the need to use the AWS S3 command line tool, but also optimize the usage of the API behind the scene. This is more or less how does it work, the schema of the deployment of this technology. You have Nextflow running into a compute node, they launch the job execution into another compute node, and most of the time this happens using containers. So there is your job, and within the same container, we operate this Fusion driver. This is the component that creates the interface between the job IO and the object storage. So there's not just this adaptation layer, this translation of API operation to S3 API operation, but also there is a caching system that make it possible to optimize the deployment on the data pipeline, the access to the object storage. So one important component that is done by this technology is to have an efficient caching using the local scratch that there is in the compute node in a transparent manner. But surely you may think, okay, but there are plenty of these tools that allow to mount an object storage into a local file system, S3FS, Airclone, Amazon Mount Point. There are many. What is different in Fusion? The difference is that we designed to make it optimize with data pipelines, taking in consideration also the data model used by Nextflow pipelines. In Nextflow, every time that you run a pipeline, each task works in its own working directory. And using this assumption, basically, we can remove a lot of limitations that there are in some file systems, because basically this model guarantees there is never concurrent access to the same file into Nextflow pipelines. So relaxing this assumption, we can optimize the allocation and the access to the underlying object storage. It's designed to run to a single job, like I was showing you before, not hosting in the compute node. And also, there is a very smart algorithm behind to prefetch all the data when you need to access data from the object storage at the same time to upload in background. So using this second level cache, they change a lot the efficiency of this system. Also, something that seems trivial or minor is the ability to support symbolic link, but this is going to have a big impact on the execution Nextflow pipeline. I will show you later why. And also super easy to be deployed. There is no moving part to be installed or services to be configured. It just requires a few configuration settings to the Nextflow configuration file. So to compare, to make more evident what is the difference between this technology compared to other S3 fused driver, we compare with a benchmark with our clone Amazon Mount Point using FIO. This is a popular file system benchmarking tool in different scenario, reading small file, reading large file, and writing files in a sequential and random manner. This somehow simulates the access of data made by the pipeline. And you can see that when you read small file, all these technologies have more or less the same throughput. The fastest one is our clone with nearly 9,000 megabytes per second. Mount Point came immediately after 8,000, and then there is Fusion, so a bit slower compared to this solution, and the same for random read. But, more than but, here what is interesting to see is that basically all these tools benefit by the fact that you can cache this file, this small file can be cached by operating system, caching system, essentially. So basically when you are reading this file, you are reading from the memory of the computer, not from the remote storage. This is why they are all more or less fast enough the same manner. But when we work with large files that are much more common with genomics pipeline or life science pipeline, the number change dramatically. You can see that Fusion can read a file like this, 6,000 more or less megabytes per second, error clone 85, and Mount Point 3 megabytes per second. So it's huge difference, and more or less also the same for random read. So why there is this big difference, and why Fusion managed to be so fast compared to other solution? For this point I was mentioning before, because behind the scene, it operates a second level cache. So there is not anymore the benefit of operating system cache, but still Fusion has its own caching system that make it possible to make so fast compared to other solutions. To be fair, Mount Point, there was a bug in the version that we use for this benchmark that's been fixed recently, so it's expected to be faster. And the same, we see also the same benefit when we write over the object storage, and why Fusion is so fast, because again, Fusion works essentially with the local scratch disk and upload in background to the search. So you are not uploading directly to the object storage. Also, a benchmark comparing. Yeah. Okay. And comparing the execution real-world data pipelines with Fusion or AWS Batch using also S3 and FSX for last threaded, the shared file system provided by Amazon. So when we deploy a pipeline, that in this case was an F-core or an NSEC, we just using S3, this pipeline require more or less 800 CPU hours to complete successfully. And using FSX is much faster because we remove all the overhead to transfer the data from the bucket to the job container to copy the result back. So it's nearly twice fast. But what is interesting that using Fusion, we can have the same performance. So the same performance using a shell file system like Lustre for FSX for Lustre. Even more interesting is the cost and impact on the cost of this technology. Here we are reporting the cost for the compute, the I/O operation, the data transfer, but also to store the data of your pipeline for 1 month. So for using Amazon Batch S3 is more or less $124 per month. The FSX is much more expensive because you need to use this separate file system only for the pipeline execution to store the data, so more or less $300. And instead using Fusion, only $69. Here it can be interesting to notice part of it is cheaper, but how is possible that it's cheaper than S3? Because we say that Fusion work on top of S3. So ideally it should be the same. There is not much sense being it's cheaper than S3 itself. As this was puzzling us, also us at the beginning, because it was not clear what it was happening. We were thinking there were some bugs, something like this, but then we realized that this is the benefit of the ability to create symbolic link over the object storage. So since Nextflow is designed to work and to reference file using symbolic link, but when you use just S3, this is not possible, essentially create many more copies of the same data during the pipeline execution. But when we have the ability to create again symbolic link, basically we did the pipeline create much less data compared to the only S3 version on the same pipeline. And we also compared this effect across different pipelines. There is huge difference in some cases. This is an F-core RNA-seq that produces more or less 1 terabyte of data when using Fusion, 90% less data. SC RNA-seq, 32% less data. ADAX-seq, minus 44. With SARIC is a huge difference, nearly minus 61% less data compared to only when you use only S3. So this approach gives you a much better usage for the data storage in the cloud. This technology is supported by support all the major object storage for the cloud, AWS S3, Google Storage, and Azure Blob Storage. We consider the version for S3 production ready. We are still working to optimize the version for Google Storage and Azure Blob Storage. This is also available both using Sakara platform, so our platform for deploying Nextflow pipeline, but also just using Nextflow command line tool is enough to configure properly the configuration file to use Fusion. And then the other component which we are working a lot recently is WAVE. It is a containerization service for data pipelines. And containers are key component in data pipelines that we use nowadays. Nextflow was one first tool using container in a systematic manner for data pipelines. It was also the reason why it became so popular, this technology. But the point is that in the last 10 years, changed a lot how we use container into our pipeline. The beginning, the idea was pretty simple to have just a container with all the dependencies into the container. And when we were running the pipeline, we were just using a container. Now pipelines are much more complex. They have many more tasks and jobs. And there is this pattern to use different container for each task. So you can have a pipeline with 20, 30, 40, 50 different containers, like RNA-seq. And maintaining all these containers is not an easy task because you need to create them, upload in the registry, modify every time there is a new version of the tools, keep online, maybe using also when you use different clouds. So yeah, a lot of overhead basically in our work. So here we want to push forward different model to think the container not like a monolithic asset that is built by somebody else, but it's something that instead we can create on demand during the pipeline execution. And it's enough to know which tool we want to use into the pipeline. The system assemble the container dynamically, maybe using a package manager like Conda or Spark. And also the idea is to make it possible to optimize the container for a specific computing platform. So if you're running this on Intel, you have the container for Intel. If you're running on ARM or other system, you can have the container for that system. This is more or less a schema that represents how does it work. There is Nextflow running your pipeline, launch the job, and when the job needs to be executed using container, has to wait for a container for that job, providing the information about the dependency that need to be installed into the container, the user identity, so can be authenticated private container registry, and also the platform where the container needs to run, so it can be optimized for a specific platform. And then this system assemble the container and upload into registry, so that can be used by the pipeline. I think maybe the most important point is that Wave is seen by your pipeline like a registry, but it is not hosting the container. It just delegates this to a real registry that there is behind. It's more a coordinator for the container that need to be used into your pipeline. We are operating this system since 1 year, and we had a lot of new features in the past month. What we call container freeze, the support for singularity, the ability to scan the container for security. There is also a command line tool. But maybe the most important part are these 2 points that I was mentioning also before, the ability to create multiple architecture container, because now it's more and more common to have platform like ARM64 that essentially the CPU said there is Mac computer also in the cloud when, with Graviton. And this provides a more cost-efficient computing capacity, but to be able to use the CPU architecture, you need to rebuild a container for that CPU architecture. That is not an easy task. So the idea is to use a package manager that with Wave, so that basically we can rebuild all the amount of the container when you need to deploy this pipeline into this CPU architecture. And along with this, there is also the support for Spark package manager. That is a package manager similar to Conda. So basically you have a collection of tools recipe that allows you to install these components. And the main difference that this package manager is able to compile the tool for not just a CPU family, but also for a specific instruction set of that computer. So there are different variants of CPU, and using this tool, basically you can optimize the compilation for a specific variation on the CPU, and they make it possible to create also the container that are optimized for a very specific CPU architecture. If you're wondering how much you can gain for this, there is this an interesting research paper that is linked here that show basically the compiling the tool and the resulting container for a very specific CPU instruction set. You can get up twice the performance compared to the generic version of the same tool of that platform. And since there is a lot of stuff, I think it's easier to see in practice how does it work, this technology. I want to show a bit more in concrete. I've used here, but the command line on Wave that you can download from the Wave repository. There is a binary version for different platforms, Linux or Mac or even Windows. So it is enough to download and run it. And you have this command line that is Wave and show you what you can do with this container. And let's say that the most basic operation to just access a container with Wave, I can use -i, it stands for image, and then I can say Ubuntu. It is give me the name of the container to be accessed through Wave. So I have this container name that I can use with Docker, like any other container, Docker run or just pull. So doing this, there is no big benefit. It is exactly the same doing on Docker let's do it. So alias d equals Docker for Ubuntu. So this is exactly the same operation. What you can see anyhow is that these containers are identical. They have exactly the same checksum independently if I download it from Wave or directly from Docker. So you may wonder why you should do this and download by Wave, because if this was a private container, it was possible to access using Wave with the credential that you have configured into the Sakara platform. So basically, this mechanism allows you to authenticate container in private repository and access using the Wave service from your pipeline in different cloud without having to authenticate independently. But surely what is even more interesting is, what if you want to add some new content in your container without having to rebuild the container? Usually, if you want to modify a container, you have to rebuild, upload, and then you can use your pipeline. But instead, what you can do here is maybe create just a script that can be my scripts into this directory. Then I create a little file that is hello.sh, where I put echo hello Symposium, like this, and then I give execute permission my script. Okay, the usual workflow should be Docker build and then upload, etc. But what I can do instead is say -wave using the Ubuntu image, and then specify that I want to add a new layer into this container using --layer my script. So what is happening that all the content of this directory will be bound to the execution of the Ubuntu container. So I have the name of this container, then I can use like any other container, Docker run, and then I can execute the script with /hello.sh. And pulling the container is running the container. I have the result. So again, here we didn't build any container, but the script was added during the pull operation and make available within the container. So what is it supposed to be better than building a container? Or here the idea is that since we were saying that in pipeline there can be tens of containers, we may need to add to all of them some script that we want to use in our pipeline, or maybe some tool that we want to use into all containers. Instead of rebuilding one by one, we have a mechanism to modify altogether all of this. That is exactly how we add the Fusion client within the container using this approach. But you can even build a container with Wave if you want. You can do just a Docker file. Like before, Ubuntu, and I want to add again the load script into user log be hello.sh, and then I want to execute on the fly. To build the container now, instead of -i, I can say -i file, and then the Docker file. And again, I need to specify this directory where I have the script that I want to add into the container. And now I have a real build. This is the name of the container. I can run on the fly. Now it takes a bit more time because here now there is the build happening behind the scene remotely. And now we have the container. Indeed, if I jump in my inbox, we find notification email that's been sent now. That's the build ID, the username, where the container is hosted, some other metadata, the image scan I was mentioning you before. So this give you some idea, something that you may need to fix from security point of view into the container, etc. So this is a way to build also the container on just in time during the pipeline execution. And but we said also we don't want to use Dockerfile to build container, but we want to have more declarative manner. So imagine that we have this Conda package manager, which provide tons of tools, and we can use this in our container, just in the Conda package, for example, some tools. by tools 230. So this allows me to create on demand a container using exactly these tools. Docker run this guy by tools. Version. We have container, it's downloaded. And okay, you may wonder, okay, but why I should use this mechanism by tools? Why I should use this mechanism if already exists a biocontainer image for each package that there is in Bioconda? Well, because Bioconda is just a subset of Conda. In Conda, there are many more tools, and Bioconda, a biocontainer only cover the packaging to Bioconda. But above all, even more importantly, because most of the time, you need more than one tool into a container image. Instead, there is Bioconda, bioimages, there is just a one-to-one mapping between one tools to one container image. So using this approach, you can train container with any combination of Conda packages. Tools, this is 117. So in this case, I can have a container with both of them or any other combination container. So this makes much more flexible how you can assemble a container for your pipeline. And so you can modify container on demand, you can create on demand with Dockerfile, you can create Conda packages or Spark packages. Also, before we were saying that we may need to use container for specific CPU architecture. For example, you may want to build a container not for Intel, but for ARM64. It's enough that you have this flag and you have a container with this tool for this different CPU architecture. Now, if I try to run this, I will get an error because I cannot run this container that is for ARM on this virtual machine that is for Intel. And this. So, but this shows that basically how you have a container for a different CPU architecture. And another thing that was mentioned before, this idea of the Fritz container. Fritz container is basically the fact, if you notice, every time that I run this, I have a slightly different version name for the container. If I do 3 times, I have this number that represents the identity, the checksum of the container content is always the same, but this part instead change all the time because this represents a unique token for the container request. And some users say, okay, this is nice, but I don't want the container to change the name all the time. It mess up the tracing on the pipeline execution for the container. So this is why we added this capability that is Fritz that basically consolidate the container with a stable name uploading to a registry that you have specified, that is bit repository. And for example, docker.io, I use my own wait demo. And basically now we have the same container, but it does not change over time. Every time that you execute this, you get always the same container name. But the same container just is frozen somehow. It does not change. It's have a persistent name that stay along the execution of your pipeline. And the last thing can be interesting to see is also the support for Singularity. Singularity is very popular when you run pipeline on-prem because Docker does not fit well into HPC environment, but Singularity has its own image format, is different way to create the specification file. Instead, using this system is enough that you say Singularity and basically create a container in the Singularity native image format, and that is hosted also into the Docker registry. This can be a bit confusing because usually you are used to use Singularity. You know that Singularity create a local file that represents the container file. But it's also possible to upload this file and pull this file from a container registry. this does not mean that Docker can run the container, but still we can store the container into a Docker registry and pull from there using Singularity. Singularity exec. The only difference that there is this ORAS :/less prefix. And this was bad tools. And it's downloaded on fly and run this container. So without having the local file, basically it's downloaded and managed by Singularity itself. So this more or less all the capability of this service. I use the web command line, but when you want to use this with Nextflow, you don't need to use the web command line because it's integrated into Nextflow itself. This is also, we added a new command for Nextflow, the TZ spec, that essentially allows you to see all the container used by a pipeline execution. For example, Nextflow spec, RNSSEC, and nav. And when you run this command, it shows for each task which is the container is going to be used. This is the basic version, this pipeline that use always the same container for all the tasks. But if I want to use Wave for this, I just modify the Nextflow configuration file and say Wave enable true. And maybe I want to use this technique that create a container using the conda information that I have associated into my pipeline task. Not Wave conda, but it's Wave strategy that is conda. And now, using again Nextflow spec, it tells me, it's 1 second, but now I have the Salmon container for RNSSEC index. I have always Salmon for this quantification step, but the multi-QC container for multi-QC step, FastQC, etc. So if it's not clear where comes this package, it comes basically from the association that I have in the pipeline code, in the module. Each module has the declaration of the conda package, and the container are built on demand using this information to the pipeline module. And finally, also to show this capability, Nextflow config to use maybe Singularity for this. I need to enable the freeze capability. I want to specify the build repository, and that is docker.io, my own repository, with demo. And then also Singularity, because I want to run this pipeline with Singularity, enable true. Okay, now if I repeat again, I will get the Singularity container for this pipeline execution created with conda packages endorsed in my container repository, my container registry. And okay, this may give you an idea how it's flexible, how the service, or also how it's integrated into Nextflow. Is automatically, basically, will pull and use this container while running the pipeline execution. Coming back about the presentation here, this service, we are released recently version 1.0. We are operating like a service in the last year. It's nearly 100 million containers, so it's quite solid. Also, we are making available as an open source technology from this GitHub repository, so you can also use, but also you can download it on your computer, or if you want to modify or contribute the code, it is possible. Also, we are making available as an enterprise distribution for a customer that want to install this on-prem to manage the container into their infrastructure. And finally, okay, let's jump into Nextflow. This is the pillar of this technology, this ecosystem. And the last year, the main work, not surprisingly, was related to make more interoperable across all the ecosystem with Wave and Fusion. So one new thing that there is into Nextflow is the support for the SPAC packages that was mentioned before. Like now we can declare the dependencies for the task, not just with conda, but also with SPAC. So SPAC can be used somehow similarly to build the dependencies in the local execution or with wave container, like I was showing you before. And also, along the same manner, we added the support for different CPU architecture for the same reason. So you can specify that a task can run for Intel processor or a specific CPU target, or maybe Graviton, etc. And then again, this information is taken by wave that create the container for this task execution. Fusion is now first-class support file system for Nextflow pipeline, as I was telling you before, can be used across CPU, cloud executor like AWS Batch, Azure Batch, Google Cloud Batch, but also Kubernetes. So this makes much simpler the deployment Nextflow pipeline using Kubernetes cluster, because remove the need to have shared file system. Also, local executor, you can even run your pipeline locally and use a remote object storage. This makes more sense, maybe apart from testing, if you want to scale a pipeline into a single virtual machine into the cloud, without you can use the object storage with the local executor. And finally, there is also the support from SLARM, LSF, and GreenDenji batch scheduler. This makes more interesting instead for the deployment of Nextflow pipeline on-prem with the object storage storage system. And why we should do this? Because at the end, also on-prem, we have very similar problem like in the cloud. Shared file system is the bottleneck for the scalability of this data pipeline. Instead here, the idea is not used natively as S3, unless we are using a Slark cluster into the cloud, but maybe to use a S3-like object storage that you can operate on-prem, like Minio or Chef, and to use as a scratch word directly for the pipeline execution. And this way, instead of using the shared file system. The benefit is this more or less the same that we have in the cloud. The object storage model provide a more efficient use of bandwidth, because it has been designed for a system which bandwidth is a scarce resource, so scale better. Also, it's much better in the handling of small files and the files metadata. And Nextflow tend to create small files, all the .command file, but also the tool, not just Nextflow. And also simplify all the automation to collect all this garbage during the life cycle of the pipeline, because you can have just some automation route to delete all these files using some tags, etc. The disadvantage is basically that you need an infrastructure that have a fast local scratch storage in the compute nodes. This come back with the idea of the schema. Does it work? Is Nextflow run the compute node? The schema is the same. The only difference is that we have an S3-like object storage that maybe can run on top of the shared file system on dedicated storage. So I think the interesting part is that we remove the bottleneck that there is in shared file system, because the job tends to use as much as possible the local storage instead of working always over the shared file system. But the access much more distributed. This should alleviate or mitigate a lot of the bottleneck into the network and the shared file system using this approach. Also, along with this, there is a new plugin for Nextflow, what we call Cloud Cache. Nextflow maintains a local database when you run it for the cache metadata. And usually it is basically run when it's distributed into a shared an HPC cluster over the shared file system. And sometimes there is some problem because not all file systems match well with this local database. Instead, using this plugin, all this metadata is not stored locally, but again, is stored into the cloud, into the object storage that we use like a database, essentially. It is also another piece to try to mitigate this problem on the access of Nextflow over the shared file system. New things that have been included in the very last release Nextflow, 23.11.0, is the support for Singularity OCI mode. So we were saying before that Singularity use its own image file format, but recently they have added the capability to run natively Docker container. Now you can run Docker container, but you need to download a transform basically into a Singularity image file. This takes time, it's not efficient, create other problem with the file system. Instead, this capability automatically run a Docker container because use behind the scene some component from Docker essentially. But this isn't nice because we remove this need to have double image format for our container and make the pipeline more portable across different computing environment. And to enable this into Nextflow, you need to specify this configuration option. Another enhancement into Nextflow is what we call topic channels. And if you ever use Nextflow, I guess, so you know that there are mainly two kinds of channels, what we call value channel that contain a single value. It can be read by many tasks. And then what we call queue channel, it is a way to create a connection between a task and other tasks. So point-to-point communication to send the result one task to another. So the idea of topic channel is to create something that is more similar to broadcasting system. You have many tasks writing over the same channel, over the same topic, and many tasks can read from there. And the main use case that originated the idea of this topic channel was this pattern that there is into NF core pipeline, in which many tasks need to send out the version of each tool, and then there is a continuous concatenation on the same channel to create an aggregation of all these versions. Instead, the idea here is now that you can create, in this example, there are only two tasks, but in any case, you can have an output, they send over this topic, this version, that you can have another task that always use the same topic, as many as you want, and then you can reference the topic channel, and this can be used any other task. So you are getting a channel that get all the data that is coming from all these tasks in a single command. Along with this, something that we are adding soon is what we call the command output type, a new type into the process definition that essentially execute a command on the fly. It can be useful to collect some metadata from the task execution context. And this again came from this pattern that is in a core pipeline. If you notice, all the tasks have this boilerplate at the bottom. So there is the real command that you want to execute, and there is always this snippet of code in all tasks. It is a bit create some noise, I think, into the definition of the task. I would like not to see this because it's not what the task is really doing. So instead of using this approach, the idea is to change and to keep the script just running the real command need to be executed. And the version of the tool that the task is capturing into this snippet instead is executed by this output definition. So basically, I'm telling the output should execute this command, and the output this command is captured then by the channel. So this make a bit cleaner the definition of this task when you need to define something like this. Roadmap for next year, what we are going to implement in the future. There are plenty of stuff. Actually, this stuff on Singularity has been already done. But I would like to focus a bit more, instead of talking one by one, on this idea of what we call DSL3, that is not going to be a new reimplementation by the language, but more a consolidation using a static type version of the language. So now there is a lot of problem that came from the fact that all the language is executed in a dynamic manner, especially when there are error messages or failing conditions. It's very difficult to capture and provide a nice feedback from what is failing. So we want to write a version Nextflow that is much more static oriented, so that there is a parser behind the scene that can capture much in a better way all the error conditions that there are in the code, the reporting in a more systematic manner, also to provide a better experience for the developer when you use an IDE, so that when you write the Nextflow code, the environment can suggest you what you can do or what you cannot do. So these are the main idea how we want to improve the language. And okay, I think you enjoyed. I hope that you enjoyed this presentation and this idea above all, which we are working to grow Nextflow into an ecosystem of tools, each of them specialized into a specific problem on the deployment and scalability of data pipelines. Always continue to keep the focus on this idea to abstract away of the complexity of data pipelines, allow scientists like you to focus on the real work and the logic of the pipeline. So thank you again for your attention. I take the opportunity to say thank you all the team working into this technology. Thanks a lot.
Thanks a lot, Paulo. Now it's time for Q&A. Do we have any questions in the room?
Okay.
So I was wondering, how do you deal with the noisy neighbor situation with Fusion? Because obviously my S3 is not designed to go that fast. If I have multiple people like trying to access Fusion, how does it cope with that? Let's say if I've got like 20,000 samples going through a BAM modification pipeline, how do I ensure it's sort of balanced and doesn't act as in a bad way towards everybody else using the S3 system? Is there a rate limiting system?
So your main concern, I didn't get what's the main concern of the BAM file.
So my main concern is that if I have a fair number of hosts using Fusion to access S3, how do I ensure, do I have to do the rate limiting at Nextflow level, or should I do it at Fusion level? Is there some kind of way of making sure they all play nicely together?
So the way I think is just a way to simplify the installation. So it's completely indifferent from the point of view of the execution of the pipeline. And then all the point is that in any case, when you use an object storage, you need to make accessible the data that is into the object storage, into the container. There is no solution to this. The only way that we can do this is copying the data in a brute way. So AWS CP and copy into the container. Instead, what we are proposing in this approach is to have this integration layer that make this data transfer more efficient.
Yeah, I was more asking if I've got like a lot of hosts doing that all at the same time, how do I sort of shape that traffic? How do I make it be nice?
Storage like S3 has no problems on scalability. Can cope, no? Maybe in on-prem is a different kind of story. Sure, you have to have, but S3 object storage can scale quite a lot.
We did kind of break it once.
If there is an Amazon representative here, we can discuss more.
Yeah, so S3 scales on prefix. So you have basically the amount of GET requests is limited by a prefix. So you have 3,500 requests per second for a prefix. So if you get into problems with parallel transfers, you should just change the way your data is partitioned in your bucket. But to get to hit those limits, you would have to try reasonably hard. Okay. So I guess following on from that, I think the core principle why Fusion has been so useful is that you delegate that sort of thing to the instance level. And so the mounting of that data into the instance happens at the instance level, which is why we need to use Wave to inject the binary into the container and then get that data in. So essentially, you are bottlenecking on the networking available for that instance to get that data in. And so that's how you can scale and parallelize the whole process, if that answers your question. With previous iterations, it wasn't quite done like that because the first version of Fusion, we were doing it at the head node level of Nextflow, and that was throttling all of the data transfer. But now, just to properly allow it to scale, we're doing it at the instance level. So essentially, wherever your task is running is the limitation.
Hi, so I have a question about HPC systems. So I work with the cluster here at Cambridge, and one of the bottlenecks I have found is the configuration, really. Because we have several partitions, there are a lot of users, and sometimes some configurations doesn't work at the time. And then if I change partition, then I need to also change the configuration. And that, as a user, has been a journey discovering exactly what I need. So I wonder if there are anything on the works regarding automated configuration, because that would be just fantastic.
Well, there is no a standard recipe also because HPC not are all the same. There are different systems. Each HPC are different kind of storage file system. But we have on the blog some tips and some guidelines how to improve the scalability. One is to try to keep a bit more under control the number of parallel jobs that Nextflow can submit. The other is, I don't know, to run Nextflow not in the head node, but as a job into the cluster.
Yeah.
The other one is to use as much as possible the stretch storage in the compute node. That I think, what I understand, is not a common pattern that you have here. So there are some tips, but depends case by case. So maybe we have to see more in details what kind of problem exactly you are experiencing.
Hello, sorry in advance because this might have been already asked on Slack. I didn't check. Is there any roadmap to make Wave standard when developing NF core modules? Because when you need more than one package, then you need to go to try to find, maybe build a multi container. If it's a package that's not in Bioconda, you have to do a PR to biocontainers, and this is a bit annoying. And while Wave just solves this issue.
Yes. So there is one. There is an ongoing PR in which we are trying to improve the capability on Nextflow to provide a module local configuration that should simplify a bit the way to define the configuration for a specific module into your pipeline. This is also related to containers. And the main idea, essentially, at least what we are trying to go, is to have just one definition of the dependencies for the module using Conda, for example, because it's the best way to track all the tools that you are using into your module just with a declaration on the Conda packages. And then using this information exactly to use Wave also to provide the container using that Conda packages. And the container can be both for Docker or both for Singularity, or because we build the container for both, or because now Singularity is also able to use the same container that is always the same Docker container. So this is the main idea in which we are going to forward. So better way to have a local module configuration than to use Wave to have a single container built from the packages that you declare into the module. Okay.
Thank you.
Okay. All right. Thank you.
Thank you, Paulo. We now have a refreshments break for 20 minutes, and then we'll be back for the rest of the session. Thank you.
Lovely. Welcome back, everyone. If you're new to the session, welcome to the first ever Sanger Nextflow Symposium. And our first speaker for this session is Nuno with predicting the protein function impact of every single amino acid substitution in the human pangenomes. So, round of applause, please.
Hi, thank you. I'm Nuno. I'm currently working at Ensemble in European Bioinformatics Institute, and I came to UK to start working here. And today I'm going to present my work that I've been doing to predict the protein function impact in human pangenomes. So basically at Ensemble, we provide genomic resources for vertebrates and model organisms. Everything that we provide is accessible through our website at ensemble.org. And the Pangenome human resources specifically are currently available in rapid.ensemble.org. So for all the latest improvements, you can always read our papers. We publish usually yearly. And you have this lovely website that you can access anytime if you want. My team specifically works on variation resources. One of the tools that we provide is the Ensemble Variant Effect Predictor, or VEP for short, that allows to annotate genetic variants and predict their consequences. So we have a web app where you can input your own variants, and we also provide the program for command line interface, also in Docker and Nextflow. And if you're interested in Nextflow, don't miss out the next talk by Likita after some talks. Currently we have a pipeline to allow us to predict protein function impact. It runs two predictors, polyphen2 and SIFT. And these predictions are based on all single amino acid substitutions in the proteome. So for each protein, for its position in the protein, we get all, we simulate all substitutions of all amino acids. And we try to see if the protein can tolerate or not that substitution. These results are all stored in serialized matrices within the SQL databases, and the results are published in Ensemble Web and Ensemble website for all users to use. So this pipeline is currently good enough for one assembly, for one human assembly, which is what we use. But with the emergence of pangenomes by the Human Pangenome Reference Consortium, we thought about redesigning this pipeline to handle much more data. So the HPRC intends to provide better genomic representation of diverse human populations. Currently, they release genomic assemblies for 47 individuals, and it's estimated to go up to 350 by mid-2024. So while redesigning the pipeline, we wanted to go with Nextflow, mainly because it allows us to share easily our pipeline via GitHub that is currently open source. And also because it allows us for multiple FASTA and GTF files, so for multiple sequence and annotation files for different assemblies to translate the transcript sequences. For this, it's good to do this in parallel, given the sheer number of files that we have. Also, a good thing about Nextflow is the built-in support for Docker and Singularity, of course, that allows us to use this in multiple types of machines. And then from this FASTA, we basically store MD5 hashes of sequences in the database to see which ones we already have done or not, and we get unique translations. So basically for each different protein, for each different human genome assembly, we can just do the unique translations. All this is made using Nextflow and Groovy, specifically the calculation of MD5 hashes, which makes it much easier to process. And then from these unique translations, we have two modes of running the pipeline. One is in full mode. Basically, we discard everything that we've done before and rerun everything again, which we do, for instance, every time there is a new version of the software and we want to discard previous predictions. But in this case of having continuous releases of new human pangenome assemblies, we want to maintain the ones that we already have and just add new entries. Yeah, so as more human pangenome assemblies are released, this is useful for us. So for every single translation, we simulate all single amino acid substitutions for that protein, and then run SIFT or polyfen. And Nextflow is really helpful here because we are running millions of jobs in LSF or Slurm, depending on the machine that we are using. And also because it allows us to use memory settings per job based on the sequence length, which also optimizes really well the memory requirements for the job. Finally, we store all these results in a database, and then we publicize for biologists to use. In the end, after running, we also get automatic email reports from Nextflow, which is also useful to see how the pipelines are running. So another good thing about Nextflow is that it allows us to integrate with other pipelines. So basically, for instance, this is a collaboration with a PhD student, René Salles from CMBI, where basically we have this pipeline that the idea is for a custom annotations from patients to see if the variant is of higher impact than when using reference annotation. So for instance, in this case, if we go to use our reference annotation, we maybe find that this variant is intronic, which means it's low impact. But if we use the custom transcriptome of the patient, maybe we can see that for the annotations of this patient, it will be stop gain in this case, so high impacts. So this pipeline, basically what it does is try to find from the sample transcriptome of the patients to predict novel transcripts. And from there, we use a Nextflow pipeline for ensemble VEP to get the amino acid substitutions for missense variants together with the patient VCFs. And then we filter out common variants. We finally use the protein function impact prediction pipeline that I mentioned you to get which variants potentially are tolerated or not. And from there, we can add again with VEP variant effects. So in the end, the output is variants that are predicted to have a severe effect. So thanks to Nextflow, we could create these scalable pipelines that handle multiple data files. Also, the built-in support for Docker and Singularity allows for reproducible workflows. It's portable, easy to parallelize with LSF and Slurm, easy to share pipelines via GitHub, everything is open source, and easy also to integrate with different Nextflow pipelines. In the future, we also want to improve some processes for really large peptide sequences, such as titin, which is an enormous protein that has given a lot of headaches, and also use this with upcoming human pangenome assemblies, as I mentioned. And in the future, we intend to publish protein function impact predictions for all the human pangenomes in ensemble, so it's free to use for everyone and easily reachable. And finally, I'd like to thank you all for listening and all the team that has helped in these efforts. Thank you.
Lovely, thank you very much. So we've got some time for questions now, if anyone has any.
So the original pipeline that you talked about, it's designed for the human pangenome, but as other pangenomes are coming out, the mouse pangenome and some of the plant ones, can that be used for that as well?
So the pipeline itself, it just needs a fast and the GTF to work. So yes, it can be used for another pangenomes. Our priorities at Ensemble right now for our team is not to do for other species, but we are considering it, and it may be in the future, but yeah, I can comment on Ensemble itself for now.
Hello. So you mentioned pipeline works on LSF and Slurm. Do you see any different behavior Or does the pipeline work equally well?
Well, I had to prepare different settings for each. I mean, I don't know if it's the way it's set up in our servers, but LSF is a bit worse. So I had to put, so the problem, at least for me, when I put in LSF is that it can handle so many jobs being put out by Nextflow. So we have to put like what I don't remember. We have to put a limit on the number of jobs that we can send at once. While Slurm handles it nicely. So for instance, it's those kinds of small things. Yeah, but yeah, they had to be a bit optimized.
That's everything, and thank you very much. And
thank you.
We'll move on to the next one. The front row is reserved for the speakers of the session. So I don't bite. You can come and sit down at the front. You don't have to worry. Right, so next we have Nathan on how Nextflow is key to the digital transformation of WSI's pipeline solutions. Take it away.
Thank you. Good morning, everybody. Very nice to be here. I'm Nathan Cunningham. I'm a director in the IDS department. That's Informatics and Digital Solutions here. We also do science. I do science as well. So we're trying to make this relevant. What I'm trying to do with this talk is just give you insight to what our teams are thinking about, how we are trying to understand how to take the big infrastructure footprint that we have here, the 120 petabytes of data that we hold in our data center that we think is going to grow to 200 over the next couple of years, take that forward with the right type of tooling, the right type of approach. So I'm just really here to reflect some of the thoughts that we've been having. And it's key as also that, you know, I'll try and limit the corporate speak. I am a director here. You all know that the real work goes on with my fantastic teams and the ideas which I'm here representing. These are not my ideas. It's my privilege to try and explain them. So, and again, what I wanted to open this talk with really, and when I say Nextflow is key, what Nextflow represents is key. How it represents that as the researchers, as you want to experience the data and actually go ahead and answer really important questions like we've just heard from the previous speaker, that H1, H0 piece. And again, it's that velocity and that speed that you can try and achieve. And to put it in a kind of a clunky metaphor, really, we're in that kind of blockbuster video to Netflix moment with our infrastructure. We have a big video library. You need a lot of stuff around it to play it. It's very good. The quality of the content is fantastic. We've established that. We've done great work over the past 10 years with OpenStack, HPC, and all the rest of it, iRods. But it's how we want to experience it. How do we make that go downstream? How do we support AI, ML loadings? How do we think about big data technologies? And equally important, what we've heard today from Nextflow, how do we get that kind of scalability into different endpoints, the areas where this community represents? It's not just working here within Sanger off the core iRods genomics data. It's where do we take that to? So one of the things that I want to reflect on today is harnessing the power of the hybrid cloud environment. Because reality tells us where, you know, that data center costs 150 million roughly. We're heavily invested over the past 10 years. We have a very, very big capital footprint, and we continue to do so. And that's how our funding is presented to us from the Wellcome, you know, over the QQ, which is around about $500 million. So we have to have a CapEx, capitalized expenditure, with an OpEx. How do we get that cloud expenditure? Right now, we spend roughly about $1 million a year in different public cloud environments. And that's tiny compared to our investments in some respects, but it's how we grow into that. And what does that look like? Where do we put the insight? Where do we put the investments? So that's what I'm trying to do today. Right. So we know we're growing under rapid transformation. Tell me an area of research that isn't across all the fields. Advancements in technology, how we look at some of the 30 kilowatt uploadings, 45 kilowatt, 80 kilowatt loadings, are things that we're investigating now in the data center. The exponential growth in data generation. I think of Matthew's here with the Tree of Life. That's going very well. That will basically be driving petabytes of data towards us as that becomes more and more successful. And that's the same for all the programs here at the Sanger. And then growth of new analytical and methodological approaches. As always, tell me a time when science has never been doing that. We always have been doing that. 10 years ago, I was the director for big data around ESRC, so hubris and enthusiasm, you could say, but actually at the heart of it is an important I/O thing there, like there is with what we saw with Nextflow. It's important about how we get that velocity. What hasn't changed, we've grown the data, we've grown the compute, but how we do the I/O, that's not changed in the same order of magnitude by any means since the 1970s, maybe a 2 or 3 rather than the 18 or 20 that you've seen in the data and the compute. And we need to stay at the forefront of that. So what we currently do at Sanger. So I put this up for 2 reasons, and one was not to wind up my teams, because this is a massive helicopter view, wrong end of the binoculars, okay? But it really just shows you the moving parts. This is basically what we've solved for and how we operate. So you've got our main devices, our at-scale sequencing operations, drives that through into the Sanger, and then we have the pipelines group headed up by David Jackson. In our pipelines teams, and essentially we have the apps and daemons around P4. We kind of got bespoke data. We've got some off-the-shelf stuff in there. That puts us into a MySQL type world with LIMS. And there's a lot of things going on here. There's Postgres, there's a whole lot of teams, there's massive amounts of, depending where you look into the Sanger, there's 80, maybe 100 FTEs doing some of this activity when it's coming through. There's a huge amount of investment in these areas. So please, this is the Times World Atlas trying to explain an A to Z of Cambridge, okay? So take that health warning with it. Metadata, through to the storage. Again, I've just called it iRods and Lustre, not to wind you up, Pete, just to get it down on there, high speed and our general store. I haven't talked about our S3, our Isilon, that's ECS with Dell. We're looking at that and all the rest of it. And then it goes into the compute side, of which we have invested very heavily and successfully produced high quality iRods product out of our OpenStack environment using the HPC posture. And that's from the on-prem. And we then have also, as I said, we're always spending in the cloud, but I'm just giving you the current view. What am I basing the current if I'm doing digital transformation, where we're going? And again, why are we doing it? Well, we all know this. This is just a highlight slide. This is, you could probably chat GPT this, in all honesty. It's pretty generic stuff. So maybe someone could tell me. No, I didn't, right? But I could have, I guess, right? Or bad, even that might have done a better job. So, but basically we have outdated software. A lot of that stuff is on Perl. It's come out of the start. We put this up 10 years ago. This is just reflecting where we need to go. How do we get that Netflix? How do I decouple the infrastructure more? And I say, how do we get the velocity that we need? Deprecated libraries, and again, reliance on some libraries that are no longer maintained and secure, technical resource gaps, inadequate documentation. Now you'll see this everywhere, right? And I've put this in. This is not us to really worry and panic about it. I'm trying to call this out honestly about where we are. So we put an investment plan together on the infrastructure, on the teams, on the IT department, so we can scale and solve the right parts of the problems for you, our community that we serve. And it's not us telling you what you need to do, and it's moving it from the computer that says no to the computer that says yes, we get that, but it needs complex orchestration. And that's basically what the teams invested in the IT department can do for the Sanger and the wider community, and stand up a system that will work to solve the next 10, 20 years worth of questions that this community is asking. So that's where we're coming from, where we're going to. So I want to leverage the strengths of private and public cloud, as you would expect, but it genuinely is a multi-cloud hybrid strategy that we have. I can't emphasize that enough. We have the OpenStack cloud system in now. We want the scalability that brings us. We want the cost effectiveness. One important thing is about where the software resource is sitting and how it's being orchestrated and where it's running. Remember, we have the 120 plus petabytes now growing all the time. We give our data away for free, as you all know. We are in the academic sector, as we all know, and that's entirely right and proper. But it does mean breaking that blockbuster to Netflix dodgy metaphor that I just stood up is we are monetizing this. We have a big video library, but we're giving it away for free. And that's what we want to do. And that's what we invested in to do. So as we know, how do we monetize the product? Well, we don't. That's why we need the capital investments. But how do we get velocity? How do we work well and flexibly going forwards in a cost-effective manner? That's why we need this hybrid. Of course, public cloud is part of our thinking and it's part of our solutions. But again, if you look at the end, that's that flexibility and scale. But the end one then is data security and compliance as well. One really key, I just don't think Vivek's here, but he's one of our first informatic leads to do trusted research environment pieces of work with Queen Mary. That's really important. How do we, as we go forward, start to bring in more of the work, say, that EBI do over the road, some of the more personal sensitive health, kind of Caldecott Guardian type thoughts? Where do we stand that up? And remember the network here, we have a very high speed network to run the scientific loadings. Then it's more of an open, what you'd see in a campus more at university. So again, it's very hard to segregate. So it needs a lot of thinking, and how we achieve that posture is part of this hybrid solution. I just want to call this out. So OpenStack is empowering the hybrid cloud environment, and it does give us that scalability, resource management, flexibility, and security. It's a CapEx investment. We can demonstrate return on investment, and we know how to develop the stack as we go forwards. But we also know, and I've used Nextflow here, and it's excellent, as just so you all know. I do really want to say to you, we understand that this offers you really useful things, just as Conda does, just as any of this kind of some low-code solution might do, and giving this flexibility like Cloud Nebula and all these other things. What I'm trying to get at is why do we want to do this, and how do we work with you to give you what you need? So part of the thinking that we want to establish is, as we look at our investment line, it's not one old block. We've got legacy, so the things that's going to get sunsetted. You've got business as usual, the stack, the thing that we invest in now when it runs now, it's running science now, and the future. So that's the innovation, the dev space. So again, part of our thinking about this is, how do we understand what this is? It does offer some challenges, but it offers a way. You can see I'm trying to call out some of the value here. Where do we take it to? And again, it's like working in that dev environment. Let's give you the environments that you need to test this, but let's not break that kind of core corpus of our iRods type of data. So it's that balance we need to make. So again, took me ages to do. I swapped out that bottom bit to Nextflow. So you can see and I put Cloud VM on the other bit. But really what I'm trying to get at here, guys, is the underpinning infrastructure as a service. What I mean by that behind that OpenStack, HPC, how we operate, that posture we achieve, is that underpinning piece. And it's really important how we layer on top these other things that give you what you need in terms of the velocity, the flexibility, and all the rest of it, and we can achieve that. And we can basically, with Nextflow, with Cloud VM, with partnerships, we can put that on top. But I am emphasizing that hybrid posture here. It's not, we're going to flip into a cloud first. And again, if you want to look at that as a community, Broad has been reflecting on some of their fantastic work they've done with Google and gone into the cloud, but they've also found it very hard to monitorize as they've gone forwards. And again, this is because, again, that corporate, nobody's trying to do anything nefarious here. It's just honestly trying to find things, how they scale in environments like this. When you are a genuine big data generator, you are genuinely one of the global big data generators for genomics. That's us. That's what we do. So we have to solve for that. So future state. So synergistic. So these kind of copy each other, these two statements. I've just pulled out different things. So what we want basically is that, what does containerization, Nextflow, complementary technology empower researchers to do? And so this is why I think we're solving, and this is what I want to basically your COI, your enterprise partner, our team, to the ask, to the PI, to the science ask, to the H1, H0 type questions. How do we stand up the right services? Automate and streamline resources, package and share resources effectively, lightweight and portable containers are part of that. Process and analyze large-scale genomics data sets with Nextflow workflows using Spark, using whatever. What I mean by that is that, how do you get in front of that containerization and start doing that analytic and that insight piece compared to the corpus of all the potential data that we could stand up at you, and the fact that we can't put it all into a container because it's bloody massive and you need a big data center to do it with. And then you need automated software tools and shared resources, so it's easy to share and work with. I've called out GitHub there, but these other things, but GitHub is a useful technology, I believe. And then we feel, I think, if these are pillars of what we're doing, and this is me trying to write kind of an underpinning piece as we might pull this together, accelerate research. Let's measure that. Are we actually being useful for you guys? Enhanced collaboration. Are we giving you tools that makes it easier? So you basically, when FTP is not enough, right? When you have to do other things. Promote reproducibility. And it's really critically important that research integrity, data reproducibility, absolutely essential as we go forwards, especially as we start thinking as we might start building out knowledge bases and do some other types of activity. How we are going to articulate our underpinning knowledge, absolutely essential that we can go right down to the base assumptions. And then lastly, because I have to think about this, reduce costs or maybe pragmatic costing, maybe should be a better way of saying that. It is a hybrid solution. It's that CapEx versus OpEx. Maybe that's again too strong. It's CapEx with OpEx is what I'm trying to articulate and work out what that balance point is that gives us all that we need. And then lastly, it's that fostering collaboration and accelerating discovery. If we're doing this right, I think that's what the future could look like for us. And I'm just trying to call out with some simple boxes there. Integration of the technologies, that's given us the streamlined containers, and it's given us that kind of speed and what that gives us. The transparency gives us the reproducibility. So what you're doing is trusted. No researcher in this room will move forward without trusting what they're looking at. Being able to publish that into a 4-star journals, obviously, every time. And a hybrid cloud. We have to own that hybrid posture. It's a Sanger thing. It's something we need for the scale that we're at, but we need to own it and make sure it's working for where we're going. How do we move, just to finish on that dodgy scenario, from that Blockbuster video model to that Netflix model? That's what we're after doing. Thank you.
Thank you very much. Any questions? Have fun.
Thank you. Oh. No, you do. You choose. You choose.
I was chosen over Paolo. Wow. Thanks, Nathan, for that talk. I was wondering with the hybrid system, with the public and private cloud, does the public cloud like kind of get access to the private system's files, like data store, or is it like kind of copied over? Is it like a copy of the
Good
data
question.
is accessible?
It gets right. So we need to mature our data product approach, open, safeguarded, and controlled. Open genuinely means open. It's our open, fair data. You always have access to that, but I'll explain in a minute. Safeguarded is really protecting the IP of the researchers sitting inside the Sanger. So they want to share, but that's under the researchers' normal working practices. And controlled means controlled. It's either on a posture from personal sensitive variables, MOD type work, highly commercially confident type work. What you'd mean, it's a controlled environment. So no, you won't have access to that unless you're party to it. But to answer your question, what I want is a set of, as we orchestrate that into, say, containers, and we talked about data, how do we think about this? We're trying to get velocity. So we're investigating certain tools. I'll just call out one. This is just one of many. We're thinking of things like Weka and things like this, how we get velocity onto the stack and how we can stand up a set of services that could look about it. What I want is basically the ability to stand up a set of information products which are got less data in them that are useful for insight. And then if you want the data products, then there will be a matter of writing versions of that, but I want to limit what that means. And what I want is a repository of published versions that have value, just like, say, the Met Office do, just like NASA do for the remote sensed imagery. How you access that? There are cuts of that. And that's what I need that to be as pragmatic as possible, because that will be the thing that you would be able to access easier. Doesn't mean we can't do other bespoke work, but there's a different chain of work there. But ideally, what I want to get to is a set of information products, the summaries of the data. They have high velocity. They are the things that can be spun around very, very easily. And that's what I'm after. Does that make sense? So that's like you accessing, again, on Met Office. If you want to look at the 70-layer climate model with the Cray £2 billion computer, yeah, you can. You can consume that on the HTML5 looking at the layers on any browser, but you can watch your weather forecast. What do you want to do? Is that all right? That answer?
Yeah, I think so.
So it's yes, but you're not going to get. I'm not going to open that up to everybody because I can't. That doesn't make sense. Yeah.
Somewhat related. How do you manage data in hybrid cloud? I mean that maybe you have local pipeline, you want to scale out into the cloud, the data is local, or you replicate the data. How you manage that?
Okay, so we internally, we stand up a set of products. So you've got Pete and Martin here. They're the guys who've architected what we do there. So we have the idea of a farm, and that gives you a set of products that we utilize around a key set of technologies. To get that into the public cloud, it depends. We would do a cut of the data and use the tool, the most appropriate tool for where we're going. So we're quite agnostic really about where it goes. So on your talk, we love all that we're saying. The thing that we need to think about the dev is, as you can orchestrate on our S3 onto our ECS or wherever we go, we really have to think about, we'd have to stand up a virtual S3 with the right type of data assets on there, and then that could flow. And it's not that I want to be putting bottlenecks in, it's not that, but it's that primary data source. Because what I would prefer, if you wanted to get hold of the corpus of high quality genomic data produced in iRods, that we build services to that basically leapfrogs the need for you to use the S3 piece and give you those tools. So I'm sorry it's a bit of a generic answer. The real answer is you need to talk to Pete offline, and he'll give you the full story about what we actually do. When we work with Biobank, the things that we've done, when we were doing the Heron piece of work for the COVID response, and how we worked as the key instigating institute, as how we responded to that. There was a strong, as you would imagine, obviously in the labs, run by the science, but it's all about the data. How does that get velocity? Where does that land? And we work with an independent company to do that. Thank you.
Hey, I'd like to know more about the granularity of your hybrid environment, or at least know your thoughts about that. Do you think it's going to be, say, all instruments that have got all their data processed in the cloud versus other instruments got all the data processed in-house? Or no, within one pipeline, like some steps to be on site versus some steps in the cloud, and having no very fine-grained ability to jump environments?
It's the latter. The reason is we have to be pragmatic. It's where do the costing comes, the other. So we're solving for data, compute, software, network, and security. That's how we deliver everything, all those postures. And we need to achieve, there's different pathways through that depending on what we're asking. So there's different answers to that. But in terms, what we want to articulate is, say for your program, what is your core data products that you need to drive insight? So we need to call those out and actually invest in those so they're stood up. They don't need regenerating all the time. And we cut versions of those. As say, maybe an instrument changes or some underpinning methodology might nudge it a little bit, or we change basically a library class or our results. Then we decide, do we want to invest in refactoring back through the versions and all that? But that's for the core. We're not going to do that for everything because we'll run out of time and resources, and we've still got other things to do. But in terms of all instruments, we need to have an active stance on all instances. It doesn't mean that we initially invest in all of them at the same time. We need to do it on that golden thread piece, the thing that's got most value. And again, if we think about why do IT projects fail, they're trying to eat the elephant all the time. 70% still do. It's that quick wins, explain the need, get the quick wins, get it working, get the golden threads working. That will build success. But it's really easy to over-engineer. Again, the beauty of why I love working in academia and why I want to be here, it's the variety of things that we ask. It is not a sausage machine, right? There's an underpinning, yes, we need the velocity, we need the data, but they all sit at different endpoints. And we must reflect that on the solutions that we make. And that bespoke tailoring, that support from teams, the embedded support maybe from a data engineer, whatever it might look like, what teams like you've got, that must exist, because otherwise you won't get to those new insights by where the science is leading us. Because again, it might not exist, or it might exist, but it's nothing to do with how we've done it here. Do you know what I mean? So I don't want to be too prescriptive. 60-40 maybe, I don't know what the number is. There's a way of us getting the scale and the cost benefit right, but that flexibility is also critical to success. Does that make sense?
Thanks.
Should we make this the last one so we can move on and let Martin have the rest?
Yeah.
Are you also thinking about making your data available for federated analysis in other platforms? And related to that, are you thinking about implementing standard APIs, thinking of GA4GH, for example?
Absolutely. That standard API is absolutely essential. I want us to reflect on that reference architecture, the stuff you guys are doing, AWS, Azure, Snowflake, what say the happening over in the States. Absolutely essential that we understand what is our implementation architecture against those reference architecture, and that we're easy to work with against those published products, the things that you want to put into the global good, those sustainable goals type questions, those other open and fair data type questions, that we need to make sure that that is being presented to the community with the tooling and the services that it needs for it to fly. Does that? Yeah.
Very much.
Thank you.
Apologies for calling you mine.
It's all right.
Your name's written on the board or anything.
Oh, I didn't sit in my excellent chair.
Oh. Really the G drive to it doesn't mess up anything.
I think so. It looks great.
So,
Right. Nope. Hang on.
okay, I don't use PowerPoint, so how do I swap it around? Slideshow. Oh, okay. It would help if I looked at the big screen rather than. It's my first time doing this. I'm going to blame that. Right, can we hide this? Actually, we can't see that up there anyway. Okay, lovely. Right, okay, so next we have Martin, modernizing Mginify, Nextflow, and NF Core for microbiome research.
All right, thank you. Okay, morning everyone. My name is Martin. I am part of the Magnify team. My job there is to keep our service up and running. So pipelines as well do most of the day. Today I'm going to give a brief presentation of how we are moving away from a current screen. You share? Screen 2. I think we are sharing. It's just
lovely. Thank you.
All right. I'm going to talk about a little bit how we are moving away from a current workflow management into Nextflow and NF Core. So, brief agenda. I'm going to talk a little bit. I'm going to talk about a little bit about Magnify. What is Magnify for those that don't know it, and then how we are adopting Nextflow and NF Core within our service. So, in this quite busy schematics, you can see what Magnify is, all the data types that we support, and the data products that we produce. So, Magnify is a platform, a resource that EBI provides for the community. Is a platform for the analysis and archiving of microbiome-derived data. So we basically provide a service to the community. Users sequence data from different biomes, as you can see on the top left corner over there. Sequence data, they submit the data to us, and we run a bunch of analysis pipelines through that data. Basically, we support 4 basic data types, which is basically meta-transcriptomic data, metagenomic data, assemble metagenomes, and amplicon or metabarcoding data. The different analysis pipeline that we have basically process that data and build taxonomic profiles and functional profiles of the data. We make the results available for the community, to the users. We also give users the ability to assemble the metagenomes if they wish to do so, which is a quite resource-intensive process. So not everyone has the ability or the compute power to do that. We offer that to the community. One thing that we do with those assembled metagenomes, for every single protein that we predict in those assemblies, we collect them all of the predicted proteins into one massive protein DB in a non-redundant way. So at the moment, it's the thing in the middle there is like nearly 3 billion proteins that is also made available to the community as like flat files on an FTP server. And the other byproduct of having those assemblies is that we bin the assemblies and we build metagenome assembly genomes, which we collect in a biome-specific way. So we have collections of those MAGs or metagenome assembly genomes, and we also provide those to the community. Everything is accessible through our web interface, API, FTP servers, and whatnot. So as I mentioned, we support the analysis of 4 basic data types, which are amplicon, metagenomic, metatranscriptomic, and this is just schematic for the pipelines. I'm not going to talk into detail about the pipelines themselves, but they will be, in a way, I'm highlighting some of the shared components just to give you a sense of how everything's built on our site. This is the third kind of analysis pipeline that we have. This is for the assembly metagenomes, and it basically covers functional, taxonomic, and sort of like system kind of way of predicting some bits on those assemblies and whatnot. This is what the output of those results look like in our website. So we present some classic visualization chart and displays and whatnot for users to go and look at what the results look like for the analysis and whatnot. This information is also available through our web API for those that do not like to click around. This is like we want to get the results of the analysis and go there, and they can use our web API for that. So as you can imagine, keeping this service up and running with the amount of data we process every week, we do pipelines a lot. Our team is basically my sub team develops and maintains those pipelines, and our job is to keep them running, processing data as fast as we can. One of the things that we basically are shifting now is we are building more and more pipelines to tackle the complexity of data we're receiving, new sequencing technology, new approaches and whatnot. So we are building many more pipelines instead of having only like subcomponents. We are building more and more of those pipelines. And I talk about briefly how Nextflow is actually helping us do that. As I was saying, this is like a punchline. Our team need to be those pipelines to be stable. That is key. And they need to be versioned. We need to keep the ongoing development efforts and the maintenance effort to a minimum because we don't have a massive team, so we need to keep things in a sensible way. And because we need to uphold the highest quality standards as we can, because we are a public service, so we need to keep doing that. So Nextflow, NF Core, and Bioconda fit this model and the mission that Magnify has quite well. But the title of the talk is Modernizing Magnify. Why? Because the pipeline that described at the beginning, they were built on using CWL, and we were using Toil as the execution engine for that. But we bump into many different issues with that. First and foremost, the stability with Toil and CWL was being a massive pain point for us. It was not behaving as expected in terms of the throughput, but also in the complexity of this pipeline. We were adding more and more tools to the pipeline, and CWL and Toil was just not working for us. The developer experience on CWL and Toil was not great. Debugging failing pipeline was a major issue. It was taking ages. And the onboarding of new people into the team was also something that was a bit tricky. So, and the last thing that I think is important when you run tens of thousands of pipelines every week is that you need to have a good, robust caching mechanism, because if one of those pipelines fail in the middle, you're wasting hours of compute time. If you don't have something that can read-write things in a sensible way, you can just like overcome the common issues from HPC clusters. So we at that point we decided to move away from it, and we say, okay, what's out there, what's people is, and then the Nextflow basically became the most viable option for that. I think there are a few reasons for that. I think the Nextflow community by itself was growing a lot when we decided to do this, and we was like, I don't know, 8 months ago. MLUAI was sort of like pivoting into Nextflow also, so that's strong in numbers. So being a small team within EBI and using that CWL kind of thing, being the odd one, the black sheep in the team, that doesn't make sense. When we were more invested in Nextflow, we were looking to NF Core and how they were doing things, because they sort of like solve one year division that we have, and that sort of like became the second Panther. And then we ran a small prototype, and we were like, okay, let's explore Nextflow, how that works, and it became part of what the category of tools that I like the most, which are the things that just work. I just like using, I just work. So that was vital for us. So just a sneak peek of how our migration process was. So there was one pipeline that was specifically problematic with Silverwell. That was this pipeline that we used to annotate and build those MAC catalogs that we have. So we need to process hundreds of thousands of genomes, and we need to run all of those tools that I have listed in the README page of the pipeline. And just with Silverwell was becoming impossible. So I decided to rewrite it in Nextflow and see if I can actually manage to produce that. And I was fairly quickly getting new MAC catalogs out there quickly, because it was just working, everything went quite smoothly. A few of the lessons for those trying to see if they should migrate to Nextflow or not. This is like my team point of view. Nextflow operators are the bread and butter of your pipelines. Learn how to use them well. I think the new training.nextflow.io is a great resource for that. That was one of the things that got me invested. Syntax are hard to spot. USB code, Nextflow plugin for that. I think, well, Paul was mentioned this morning that's going to happen that side. And then I think I've seen most team going into, okay, we need to use Nextflow because it's the thing that everyone uses and it seems to be working. And then, but they step away from NF Core because I think most people fear there's like too much complexity on that. But I'm after going through that process, I'm going to say that NF Core has a bunch of best practices condensed and distilled into code, good practices and documentation. So I strongly recommend everyone using Nextflow to sort of like peek into NF Core and see what they can use from there. So to wrap up, by adopting Nextflow, NF Core, and Bioconda, by just sort of like using NF Core, we are in a much better position to deliver our service. We can onboard new people quite quickly now. They become productive in just like days. And then what NF Core is bringing as a software engineer myself is that NF Core brings all the modern software development practices into the bioinformatics side of things. So we have a research team. We take those pipelines they develop in a so like researching in a way and just like make them production ready quite quickly, following those practices. Finally, I would like to thank my team. The poor guys have been hearing me part about Nextflow and NF Core for the past 8 months. Thank you guys. And that's it.
Thank you. So I think we've got time for one very quick question, if anyone has one. If not, I'm sure you can catch mine outside for lunch. No? Fantastic. Anything online? Nope. Cool. You're off the hook. There we go. Lovely. Right. So next we have Samuel with PGSC Calc, a reproducible workflow to calculate polygenic scores.
Thanks. Hi, everyone. I'm Sam. I'm an assistant professor at the University of Cambridge, but I'm also a visiting researcher at BPI since 2019. And yeah, so today I'll be talking about polygenic scores, and it probably makes sense to first jump into what are polygenic scores, but for those who don't know. Okay, so polygenic scores are basically a way for you to calculate your genetic predisposition to any disease or trait that's heritable. And so how that works is basically, usually we do these studies called genome-wide association studies, where we measure all the variants in your genome, and then we look at, for each like copy of a variant of allele, how much does that change your trait value or your predisposition for disease? So then we do that across the whole genome. And what a polygenic score is then we take the variants that are associated with disease, we sort of pull them out, and then we basically aggregate those into a joint prediction of your genetic predisposition. And that's basically just a linear combination of how much that change in your DNA affects the trait value, and then how many copies of that allele, that genetic variant you have. Then you do that, you sum it all up, and that's your polygenic score. And for this scientist, their polygenic score for this this trait would be like 1.49. Okay, but that's just like a random number, and what does it do? Well, actually, these polygenic scores, if you look in a population of people, stratify them into sort of levels of genetic risk. And so, in a population, you get this normal distribution where some people are at the low end, most people are at average risk, and some people at the high end. But for traits and diseases, actually, this has like a big impact on your lifetime risk of disease. So here you can see that the people in this red part of the distribution, their polygenic score is highest for coronary artery disease, and their risk actually accumulates much earlier than people who are at average or low risk, which means it provides an opportunity to, target prevention. And that's just a general case, like for lots of different age-related disease, like Alzheimer's, different cancers. Okay, so at EBI, the main work I do is to develop this catalog called the Polygenic Score Catalog, and it's an open database with all the polygenic scores that are out there that are open and available to use. And so, it has information about how the polygenic scores were developed, who they were developed in, how well they've been reported to work. And then we provide the scoring file, this list of variants that you need to then go out and calculate a polygenic score on a new sample. Okay, but that doesn't really go far enough, because actually the tools and the expertise you need to calculate polygenic scores is quite challenging if you're not an expert geneticist. And really the power of polygenic scores will be about how clinicians and sort of less technical researchers can interact with them. And so, you would have really needed to do a lot of scripting, a lot of mapping, a lot of work with schedulers and stuff like that. And still, it wouldn't give you a good result. It would give you this sort of scaleless measure. So that's why we developed PGSC Calc, or the polygenic score calculator, and we chose to use Nextflow to implement that, because we wanted to basically abstract away all these really technical details into a reproducible pipeline that's can also be customized with lots of different parameters, but that basically abstracts away all these technical details and gives you the best practice to calculate polygenic scores. So basically there's, it basically can take any sort of target genome. So the genotyping data usually comes from a genotyping imputation, but it can also come from sequencing data. And so the pipeline allows inputs VCF files or the Plink files. Then the pipeline can either take polygenic scores from the catalog or your own polygenic scores that you want to calculate, and basically does all this technical variant matching, scoring, and aggregation, and outputs a report and the polygenic scores for you to use in your work. The pipelines are written using the NF core template, and then we test it to make sure it works with Docker, Singularity, and Conda in Nextflow. So many people just always ask us why not use Plink. That's the thing that people always use, but actually that still is really challenging for most users. And so we basically, and it's not scalable. You still have to script it all together, and that's basically what the pipeline does. It works with as many polygenic scores. It can calculate thousands of polygenic scores at once, and it works sort of the same for one sample as it does 500,000 people. It has sort of a simple command line interface to use. Basically, you input the data about where to find the genomes, what build they're in, and then which polygenic scores to calculate. It does everything, and then outputs a report that basically has information about how the score matches your how the variants in the scores match your genome, and it's all in a log. But actually, the big problem with polygenic scores is that they're sort of confounded by genetic ancestry. So this is just a graph showing you that, depending on sort of your ancestry, different ancestry groups like European ancestry populations or African ancestry populations will have these stratified polygenic score distributions that are really not informative. And so polygenic scores are on this like scaleless axis that's quite hard to challenge. So what we need is a method that actually goes into those distributions and reports your relative risk irrespective of your genetic ancestry. And there were no real tools to do this before. So that's why we built in sort of this version 2 of the polygenic score calculator pipeline that you use with this run ancestry parameter. But basically all the data handling and variant matching are the same and the scoring remains the same, but we add new functions that using a reference panel of like sort of reference populations, you can basically identify the overlapping variants and do the PCA and scoring by matching. You do the PCA to make sure that you understand the ancestry of the people you want to calculate their polygenic scores on, and then apply these adjustments that actually remove the effect of genetic ancestry. And so now the new pipeline outputs a new report with information about genetic ancestry. So we project individuals, and you can see the sort of open circles are the query samples, the target genomes that you've added, and the dark circles here are the reference populations, which is a merger of 1,000 genomes and HGDP. And then you get information about the score distributions, these polygenic scores, and basically what we've implemented is 2 methods to basically make the distributions across ancestries overlap and not be so stratified, and then also report polygenic scores on this interpretable scale, basically your relative risk of a disease based on your genetics and based on people like you. And so I'm right on time, but yeah, this is the work of a larger team based at Cambridge, EBI, Baker in Australia, and funded by HDR UK as well as NIH and multiple other funders. But thank you for your attention.
All right, thank you very much. Any questions?
How do you estimate which is the ancestry of each sample?
So we do it 2 ways. Basically, we use this like reference panel of populations. So like 1,000 genomes and HGDP, you merge them together, and those have sort of established population labels, like based on sort of the way they were sampled was to sort of have those ancestry labels. Then what we do is basically just you derive a PCA on those people and then project the new samples into that PCA space.
How do you choose which SNPs to take?
That's just using the standard. You like prune to only common, prune to a small set of common variants that have a high math. So like, and then you can either use random forest or distance-based metrics to assign population labels. But actually these are quite, in polygenic score analysis, we're trying to move away from that. These are actually very problematic to assign labels to people. And so actually the better methods now, which are implemented in the pipeline, use a continuous mapping of ancestry to decorrelate the polygenic scores from ancestry and put people on an interpretable scale. So we actually don't need to assign ancestry anymore to reduce the effects that you see of ancestry stratification.
And how many principal components do you take to calculate this distance?
So in the ancestry decorrelation, usually people use 4 or 5, but you can customize that within the pipeline. It's not really a settled science.
Thank you very much.
No worries.
Well, we're going to have to stop there with questions. That's right. Sorry, you can catch them at lunchtime for any more. Right, and finally we have Disha, migrating ensemble pipelines to Nextflow for enhanced production-ready workflow.
Morning, I'm Disha. I work in Metazoa team. So my topic is migrating ensemble pipelines to Nextflow for enhanced production-ready workflows. I work in Ensemble Metazoa, which is an open access platform to non-vertebrates genomes. And then we further collaborate with ViewPathDB to provide support to key hosts, vectors, and other pathogens. This is an overview of our workflow. We basically get some requests for genomes in terms of RT tickets, which then we download it from INSDC, and then prepare the pipeline and standardize the files. And then we load it into our CoreDB using our genome loader pipeline, and then perform various different processes, and then perform a data check and dump it back to ViewPartDB. So to further then focus on the productivity and flexibility, we moved from eHive to Nextflow. eHive is a workflow that is used by EBI right now. So we are moving to Nextflow, and these are the 3 pipelines that we have moved to Nextflow. We have been using it for 2 release cycles, that is in the time span of 4 months. And then further to tackle with the issues, we have introduced logging and testing into it. So logging has helped us in better debugging. Logging is a Python library, which has different levels like debug, info, warning, error, critical, which can be used to log the runtime messages. So you can also get a log file using log file path and the path to the file, and also the levels you would want for a log file. So with this Python library, we use the functionality of Nextflow, that is with trace, to trace the log file, because the log files are present in the work directory. So we use the hash to trace the log files. And then we also use Nextflow log to look at a process if we want to look. And we have created a template for our log file, which we use by template. And here you can see that some of the warnings that we could have got in our Nextflow pipeline while running it would be like checking for genes to merge and stuff. So this is something we want to track and look at later if there are some issues. So, and then next, for continuous integration, we use testing. Testing is also done by PyTest. So in PyTest, you require a YAML file, which is in terms of you give name, which has the name of the workflow, then command, which needs to run, and also files. So files contain to the path for the output files, which we expect, and then you check them by ND5 sum. So this is how we continuously check all the changes that we do into our pipelines and stuff. So this is all done by my team and me. So special thanks to them and extended ensemble team, EBI team. And that's it. Any questions?
Thank you very much. Any questions?
I can ask one if you want. Because you're using PyTest, I was going to ask, have you looked into the NFTest plugin that's recently been...
Oh, yes, sorry, I forgot to mention.
Oh, did I?
We did look into NFTest, but our repository it's not fully complying the NF core repository. So, I mean, you can scan this and look into our repository, but that's why NFTest wasn't working for us. So we just used PyTest for now.
That's nothing else, then thank you very much. Right, so I'm sure many people are waiting for the next part of the day. But first, there is a photo to be taken outside, and then you can have some pizza. And if you don't like pizza for whatever reason, there is some salad. But that's it. And then after lunch, we will have a talk from Harshal. So goodbye.
That's great. Hopefully everyone can lip read. Sorry. So yeah, if you haven't already started the Nextflow repo, please can you do that for us? It will help with grant applications and that sort of stuff. We've recently submitted a CZI grant application, which we've had for a few years now, and it really helps to hire developers to help with the community and to keep things going on that front, as well as sponsoring hackathons and various other things that we're doing. Okay, so a lot of you have probably seen these famous tube map representations that actually came out of the NF4 community that are now being used everywhere. And so this is what a typical pipeline looks like. It's a series of steps that you perform in a containerized way. And Nextflow really manages a lot of the complexities behind the scenes for you, where it runs these with containers, with pre-installed, with whatever you need to run these particular tasks. But also it has a number of other features like flexibility, scalability, various other things that you can exploit in order to run these pipelines virtually anywhere. So you write a pipeline once, you do it properly, and the idea is that you can share and run that pipeline wherever you want. It has a number of features where you can write code in any language and still use that within your pipelines, as well as I mentioned, using containerization to really deal with dependencies. You know, gone are the days where you have to do an install of a tool on a HPC, and it makes it impossible to share scripts in a reproducible way, you know, across the planet if someone wants to actually get the same results. Now everything is containerized, everything's run in a container. It's a really nice, compact way of running your pipelines. But then also, I think what's really changed the way that we do science in general, in terms of bioinformatics, is version control with Git. And it sort of brought in software engineering practices into bioinformatics that weren't probably there as much before, that really force scientists to think about what they're doing with their code, how they're writing their code, how you can get others to review your code. And some of this stuff actually is also pushed us in the NF Core community to implement some of these things within the community as well. So we can exploit these features and actually start writing code in a nice, open, reproducible way. But reviewing is incredibly important. And then to top it off, you can run these pipelines virtually anyway. I think there's been some conversations and chats about that. You write a pipeline once and then run it anywhere. So very quickly, numbers, Nextflow is growing, you know, pretty much exponentially. If we look at, you know, a standard metric like how many people are accessing documentation over time, it's just going up and up. You know, 16,000 users in December, which is just phenomenal. If we look at NF Core, almost 900 contributors contributing to NF Core GitHub repositories. Again, phenomenal. If we join up Twitter followers, it's almost 9,000 on for Nextflow and NF Core. The Slack organizations, we join them up, you know, 11,000 users. I joined NF Core when there were about 20, 30 people on Slack, and we were literally just building everything up at that time. And now we're almost 8,000 in the space of like 5 or 6 years. And it really sort of filled a gap for bioinformatics analysis research, not only for those that are bioinformaticians, but also those that just want to use pipelines. They don't want to have to sit there and write a complex RNA-seq pipeline. And you know, some of you in the audience here, you know, all the amazing talks that happened this morning, you write a pipeline that fills a niche. You're the domain experts, and anyone can come in and use it. And it's just amazing how it's just grown out. You can have conversations about your pipelines, various other things directly on Slack, and get help, and also, you know, stay around if possible. That's what we want. Yeah, so this is just a plot of this Slack membership over time. It generally peaks at hackathons and that sort of thing, but you know, almost 8,000 is just amazing. The Nextflow Slack now, which is a separate workspace, that we started a bit more recently. It was on Gitter before that. And even that is just, I think we're almost 2,000 members on there as well. Okay, so see, these are just some other numbers. 7K over Slack, 7K Slack users, over 40,000 commits, 16,000 pull requests, 7,000 GitHub issues. These numbers just are staggering. And then they keep on going up and up every time that we give these talks, that they're outdated. So what is the core content on NF Core? I mean, a lot of it is the pipelines. So the whole philosophy behind creating the NF Core community was that very early on, before Sakira even, at a community conference for Nextflow, a number of people decided that they would like to use Nextflow to create this resource of pipelines that were best practice pipelines that anyone can come in and use. And rather than every individual lab having their own version of an RNA-seq pipeline, they wanted to contribute and collaborate on writing a single pipeline that everyone can use. And so this is actually one of the core guidelines on NF Core, that you can only really have one pipeline per data type. And so there's only one RNA-seq pipeline, there's only one ChIP-seq pipeline, and various others. And over time now, we've really built up, it's almost like a big brain of domain expertise that we've built in this community, because I know nothing about proteomics, but others do. Yasset, we were talking to him just outside, they know about proteomics. They've contributed pipelines to NF Core. And so others that want to use these pipelines can then come in and do that. And so we're now really starting to see a shift from, just genomics type research to having proteomics pipelines, image analysis pipelines, astronomy pipelines, various other things. And in essence, anything you can containerize, you can write an Nextflow pipeline around. So it doesn't have to be even life sciences, to be honest. You can do anything in a container where you have software available. You can use Nextflow to do it and run that pipeline wherever you want. And so you can come in and just use these pipelines. And as I said, 90 pipelines. It'd be amazing to get to see us get across the 100 mark there. This is a bit what the landscape looks like. So this is a word cloud of GitHub stars across NF Core pipelines. I think this is probably outdated now, but just to give you an idea, most of the pipelines there are green because they're genomics pipelines. Then you've got red ones, which are proteomics pipelines. You've got a yellow one, which is a pangenome pipeline, which is becoming sort of the cool kid on the block when it comes to analyzing sort of human genome data. And actually, we had Adam Philippi give a keynote at the Boston Summit, which was awesome. But we're also seeing a lot more image analysis pipelines. Image analysis is a completely different type of data because it's huge, or it can be huge. And you can't open it on a laptop to start identifying cells and circling them. It's virtually impossible. And so what you need to do is you need to be able to use good containerized software to take that image analysis to a series of steps. It's also very difficult to move around. It gives you a different problem to running 20,000 small tasks. This is like running, a few huge tasks. And, how do you provision the storage? How do you run that, whether it's in the cloud or locally on a HPC? It's a completely different problem. And so this is what the landscape looks like. They've also, as I said, they've been the sizes here represent the number of stars per repo. And so the RNA-seq pipeline is clearly the most used pipeline, it seems, the most starred. And that's mostly also because there's just a ton of RNA-seq data knocking about. A lot of people do RNA-seq experiments. And so it's there. It's also probably the gold standard NF Core pipeline that we've got. Everything that we sort of implement and develop on RNA-seq, it tends to be done there first as a proof of concept to ensure that it's working before we roll it out more widely on NF Core. These are the typical components of a pipeline by our definition on NF Core. When I say our, I mean with my NF Core hat. That's so a community type hat, not necessarily a Sakira one, even though they're quite interchangeable. So here a pipeline is an end-to-end pipeline that runs some sort of analysis. A sub workflow is just a series of steps or modules that we call them that allow you to chain together some tasks. So doing some adapter trimming and then running FastQC after that, and then maybe doing something else after that. So you've got like 3 or 4 components that form a sub workflow. These sub workflows are also amenable to be shared across pipelines. And so the smaller and more compact you can make these to make them shareable, it just means that you can more efficiently implement your pipelines at scale. And then underneath that, we've got modules. So a sub workflow is formed of a series of modules. A module is a single unit in our definition of Nextflow, which is just a single process. And that would be a process to run, say, BWA-MEM or FastQC and so on. And the fact that these are all individual, again, it just means that it makes it a lot easier to share. I mean, we had to come up with a way of really adopting DSL2 syntax of Nextflow on NF Core. And this is what we eventually ended up with, is having a repository of core modules that we can have in a single repo. Contributors come and contribute those, update them, and also be able to install them in any NF Core pipeline, any Nextflow pipeline as well. So over time, we've come up with some standards for how we implement sub-workflows. They're sitting in the NF Core modules repository. We've got 54 of those. And now we've got almost 1,100 modules on NF Core, which is, again, it's just amazing. Lots of people are coming in, contributing these modules for their own pipelines. They're reusing them, they're updating them, they're identifying bugs and issues and fixes and pushing them back. We're also updating the tooling and various other things on the NF Core front to keep them up to date and to make sure that we're at the edge of using all the best sort of software implementations and practices and that sort of stuff over time. So I started this repository maybe 3 or 4 years ago at ISMB in Basel, and it just sat there doing nothing for a year. And then Paolo pushed out DSL2, and we decided we had to do something about it. And so we first implemented a prototype DSL2 implementation for RNA-seq. With some horrible syntax we used in that, but it worked. And then over time, it's just evolved and got better and better and better. And now, you know, we're sort of at 1,100 modules, which is, again, it's just phenomenal. So everything I've been talking to you about has been mostly what we're doing on NF Core, but there's also a lot of usage outside of NF Core. And there are a couple of talks today, actually, also where, you know, you guys are developing your own pipelines and you're using the template, or you're using the tooling, or you're using various other components because you just identified that it's easier to use and manage your pipelines in that way. And you can also adhere to some sort of standard internally, so you don't have to make your own up. And that, again, it's just amazing seeing those talks this morning and how you're using all of these things. And so if you do a quick search of GitHub, for example, and look for some things that are quite unique to the NF Core pipeline template, there's over 1,000 pipelines on GitHub that are using this. And this is just GitHub. This doesn't include Bitbucket and GitLab and all of the others that are out there as well. And so actually, just following on from that, what we're trying to do also, as I'll probably tell you a bit later on in future directions, is to make the NF Core template more accessible, simpler, easier to share. It's been commonly requested across the community. You know, people want to get the NF Core goodness, but they don't necessarily want all of the boilerplate, and that's understandable. And so what we're trying to do is to try and make that a bit more fine-grained and tunable. And so whenever you update or create your pipeline, you can select what you'd like to do. So it will be more opt-in and easier for you to then build on top of that and start writing your own Nextflow pipelines. And so I think that's a work in progress right now. So NF Core pipelines also, you know, are being used everywhere for reproducible research. And that's one of the main advantages. Science has to be reproducible. Otherwise, there's no point doing it, really. And so NF Core pipelines are being used in all sorts of papers. And I think the thing that I love the most about this is the fact that when you write your methods section, it can just be a single line. So I've used this pipeline with this version, potentially some parameters, and that's it, right? Because everything behind the scenes is actually managed by containers and various other things that are already pre-bundled with that version of the software. Before NF Core, you know, I was having to reproduce public data analysis, and it can be a nightmare. You have no idea how someone's run a pipeline, what parameters they've used. It's very light on the details. And so trying to reproduce those plots, summaries to write your own publication at that time, you know, was horrible. And this sort of helps with that quite a lot, providing you've used the same command and have access to the same input data, which most publications now ask you to do anyway to submit to public databases. You can reproduce what you've done and hand it off to the scientists to actually focus on, you know, what they want to add as the rest of their publication, which is just phenomenal. Okay, so the tooling. As I mentioned, we've got some guidelines. These are almost sort of the 10 commandments of NF Core, and we have some standards, some guidelines that we implemented and wrote, and they're evolving, to be honest. But the idea is that it really helps to set some sort of spine in terms of how users in the community interact and build their own pipelines. And I think it's really nice to have those. Otherwise, you end up with loose definitions of things, and you know, there's not enough clarity on what you need to be doing and how you need to be doing them. And so we were very strict about these standards initially, and that was for a reason, but actually very few people have complained about them. They see the standards and they're like, okay, well, we want to write our pipelines in this way as well, because they see the benefits. And it's awesome to you know, to be able to see that in person. So I guess one of the key ones is, as I mentioned, you know, not having more than one pipeline per data type, but also the way that your pipeline is written. It comes with standard test data. It's containerized, and so that it can work on virtually any platform if you like. It's tested on virtual platforms. We've got CI/CD as well, which is also potentially sort of a guideline that your pipeline works before release via CI/CD. Ideally on clouds as well. We've got credits from cloud providers because they are also seeing massive usage of NF Core pipelines on all these different clouds because they're just easy to use. And so I guess following on from that, that's another thing that NF Core has really changed is a lot of people are transitioning to cloud, and they have been doing that over the past few years. Big pharma, small pharma, academia, all of them are transitioning to the cloud. And inherently, Nextflow and NF Core pipelines, in the way that they're written, work on all these cloud providers, which is great because now companies are moving to the cloud and they're like, well, I need an RNA-seq pipeline. Where am I going to get that from? And so they look at NF Core and they're like, okay, we can just use this out of the box. And so now we're seeing a massive shift where all of this open source contribution now is paying off in the way that it's changing the mindset in the private industry as well, because they now also want to contribute back to open source rather than having everything gated off, which is typically, you know, how potentially things were done at that time, rewriting their own pipelines, having their own bioinformatics and that sort of thing. Now we're seeing our customers wanting us to contribute back to open source and allowing us to do that, which is absolutely phenomenal. Okay, so in terms of how you use what we've got on NF Core, there's pipelines, there's sub-workflows, modules. You can start stitching your own pipelines together. There's various linting options as well, so that we know that the template and the pipeline you're writing is adhering to best practices. There's tooling and also this parameter schema that allows you to update your pipelines. But behind the scenes, a lot of what's going on NF Core is via this NF Core tools package. The reason we need that is because we don't want to be going to every 90 pipeline and submitting a pull request to update one portion of the template. It doesn't make sense. So the only real way that we can attempt to automate a lot of what we're trying to do across 90 or 100 pipelines is to have some sort of tooling that we implement and we developed. And so we've got this NF Core tools Python package, and it allows you to do various things, whether that's creating a pipeline template or creating a sub-workflow template or creating a module template or linting or downloading containers because you want to run in an offline environment. All of this is pushed to this NF Core tools package. Whenever NF Core tools is released on pip, what happens is NF Core tools itself contains a skeleton pipeline template. Behind the scenes, what happens is when NF Core tools is released, there's a sync mechanism that gets triggered off, and it automatically creates a pull request to 90 pipelines in the same go to now update that pipeline to the latest version of that template. And so this way we can stay up to date. Whenever NF Core tools is released, we have one point to update and it gets rolled out across all of these different pipelines. And it's just a really nice, easy way of interacting with these things. But there's various other commands here as well that I'll show you in a sec. Wave was mentioned earlier. And actually, we do have a pull request on NF Core modules to add CI/CD to actually build those mod containers via Wave. We just need to figure out some of the details. It's very important to get things right. A big advocate of doing things in a small way and then rolling it out on a larger scale, because then you can try and iron out a lot of the issues that you're seeing there. But I guess one of the key things that potentially also we need to think about in the NF Core community is how are we environmentally responsible for some of the stuff that we're pushing out on NF Core. It's all well and good building 1,000 containers, but that has an environment impact as well. And so how can we be better with our CI/CD? How can we be better with the size of containers? So, for example, what Wave does behind the scenes is when you have a Conda recipe in your Nextflow process, what it will do is it will, and you decide you want to build that container with Wave, what it will do is it will use a micromamba base image, and it will essentially just do a Conda env create behind the scenes that you don't see within a Docker file. And that Docker file then gets pushed up all transparently via Wave, and then you can use that. And so the idea would be then on NF Core, we could do something similar. Most modules have a Conda definition. Why can't we just use Wave for that? But I think we need to figure out how we do that in a sustainable way. So we've been discussing potentially having our own registry where we can push up these containers, but also doing a bit more profiling of the size of these containers. Are we able to make these containers smaller? If you think about how many times the SAMTools container is pulled globally, maybe even just down to NF Core pipelines, it's probably some phenomenal number, thousands, tens of thousands. And if we can shrink the size of that container by, I don't know, 20, 30 megs, whatever it is, it makes a massive difference running these, especially in the cloud, because every task you run in the cloud has to be in its own virtual machine. And so for each of those tasks, you have to pull that container locally before you can run anything. And so we just need to, I think we just need to be a bit more aware about how we're having an impact on the environment to not be wasteful, essentially. And also part of that has been also thought about in the implementation of the NF test that we've been doing that I'll talk about in a second. So you can install this. It's available on pip NF Core tools if you just do a help. There are various options there, not only for users, but also for developers there that you can go and have a play around with. So we've intentionally started to now make a shift in the way that we're developing our tooling so that it also supports custom pipelines. As I mentioned, and as we saw this morning, there are custom pipelines that are being implemented and developed with NF Core tooling, whether they're on GitHub, Bitbucket, GitLab. And so part of that is adding the tooling that supports all of this. So for example, you could have a completely custom pipeline with all NF Core components, or you can shift out and just have a few sub workflows that you want to install from NF Core and maybe a module or two. Or you could have a completely custom pipeline. You could have your own modules repository, your own RIP mirror of that, and the tooling itself should resolve that and install what you need within your pipeline. And we know it's being used, but we haven't had much feedback. So if you are doing this and you've got any issues, then please let us know. We'd love to, have some proper use cases where we can implement and improve some of the stuff that's going on here. But it's entirely possible to do things this way. There's also widespread adoption of the tooling and various other things that we're doing and advocacy, in fact. So Backtopia, for example, it's a huge pipeline that's just written by someone at Robert Petit, who gave a talk at the Boston Summit. And it's got tons of components, and it's his own custom pipeline, but he's still using a lot of NF Core modules and contributing back those modules for everyone else to use, as well as sub workflows. There's a Tree of Life project here that we know is, using NF Core components in their own implementations. ONT are using the schema that we developed on NF Core and various others. And Element Bioscience is also a massive advocate and also are quite heavily invested in the NF4 demultiplexing pipeline for their platform. Right, so NF test. There's so way back when we started implementing NF core modules, we needed a way to test that the modules worked. There's no point adding 100 modules to a repository that don't work, because then you have no idea whether when you install it in the pipeline, it's just going to break the pipeline. And so we needed a way to test those. We needed unit testing. And so PyTest workflow at that time solved the problem. It was great. You could write standard YAML, and then you can update and implement that. Someone creates a pull request, the CI/CD is automatically triggered off on the pull request. So whenever someone makes an update to that module, if it breaks, then you know you have to fix it. And so it's a really easy way for users to get onboarded, not only just bioinformaticians, but also core users that want to write a module and are just starting to write their Nextflow pipelines. Is a fairly easy way to keep on top of it. But it has its own limitations, and it's not Nextflow native. So for example, you can't test functions and various other things that are written in Nextflow or in Groovy. And NF test came about, can't remember when it was now, a year or 2 years ago maybe, where they really sort of announced it. And very recently, we've been working with the authors of NF test, who have been really receptive and have taken on our feedback and suggestions and implemented a bunch of stuff in NF test, which is great. And what this means is now we can start using NF test as our testing framework. There is a transition going on at the moment where we're shifting from PyTest workflow to NF test. There was a massive effort at the hackathon, and there will be, I suspect, over the course of the next year or two, shifting that. But not only within modules, but also within pipelines. And so at the moment where we've come up with sort of what a skeleton pipeline would look like with NF test in it, and we're just figuring out now how we do it. But a very key thing, going back to my point about environmental impact, is if we have NF test in all of the components in your pipeline. So right now, the only test that we include in NF core pipelines is the end-to-end pipeline test, essentially, that runs all of the processes to make sure it's working. But if we can ship all of the tests that the pipeline performs by default, whether that's the modules, the sub-workflows, and the workflows, we can start doing clever stuff with CI/CD where we can essentially check for changes. So we only run the tests that have changed. Rather than rerunning everything all of the time, we can be a bit more selective about what we're running. And again, it just means that, you know, someone's not paying the light bill, and also we're being a bit kinder to planet Earth. So this is still a work in progress. The hope is to have an RNA-seq release out soon-ish with that. A lot of my team at Secura, as well as in the community, have been working on pushing all of these NF tests to the components that are being used in RNA-seq so we can get a proof of concept together. Fetch NGS actually has already been released with a lot of the core concepts that are being used in NF tests. So yeah, worth keeping an eye out. There are various other plugins. The NF validation plugin actually also came from NF core. We developed JSON schema to represent parameters that are used in pipelines, and now it's being used all over the place for various things. Help text generation, pattern validation, sample sheet validation, and it's a really powerful way of just keeping on top of the parameters that you're using in your pipeline. Also, because you can now start rendering these JSON schemas in user interfaces. So at the Epitomean Oxford Nanopore platform, that's what they're using to render their particular user interface. And that's what we're using also on the Secura platform as well, to render that platform, to make it easy and open up the way that people run Nextflow pipelines. You don't have to run Nextflow pipelines via the command line anymore. You can now start using user interfaces to run them. It's your choice whether you want to use command line or whether you want to use a user interface, or whether you just want to trigger them automatically. You don't need to know. Data hits a bucket and you just want to trigger a series of events that then gives you some sort of data at the end that you're interested in. The NF core website has been built from the ground up again. Matthias has been working around the clock on this, and now we've got a brand new NF core website, which is a bit more scalable and removes a lot of the hidden gems that Phil had in the old website. So yeah, that's awesome. In terms of the tooling, we've recently improved the download capability, and We've added also partially as a result of this, a new Nextflow command called Nextflow Inspect, which allows you to look at the containers that are in a pipeline. We had some horrible regex code that was parsing Nextflow scripts that is incredibly fragile, and I wouldn't ask anyone to even look at what that regex looked like. But we needed something a bit more Nextflow native. And now we've got that with this new Nextflow Inspect command. It's also compatible with Wave, and there's various other features that you can get in there as well. There's also a user-friendly terminal app that Phil has been helping to write, which brings this new tool called Textual in, and essentially it's called a TUI. So rather than a user interface, it's a terminal user interface. And so rather than typing out your command, you get prompted via terminal type interface to enter things within there. And again, it's a really nice way of interacting with the terminal. Okay, so very quickly on community initiatives. There's a podcast, if you didn't know already, called Channels. And I think these run every few weeks or something like that. It's worth keeping an eye on these. There's always sort of new content and new discussions around various things that are happening in the Nextflow world, and new features and various other things that we're adding also maybe also to the NF Core community as well. We have a new ambassadors program on the Nextflow side. Priyanka's part of that, and she's done an awesome job organizing this, got us all here, which is amazing. And so if you're interested in becoming an ambassador, you can find the details on the Nextflow website. We also have a new community forum. So the plan is eventually to move Nextflow and even multi-QC and various other things into this discourse type format. And the main advantage of this is that it now allows you to Google for things, which is huge. Everyone Googles for things. In Slack, you can't necessarily do that. But also there's an advantage to using something like Discourse because it allows you to scale. And given the size of these communities, as I showed you earlier, we need something that is actually resilient to that. And so we're trying to come up with a unified way of actually communicating. And so please check it out. It's I think it's community.secure.io or something like that. There's more details. Also, Phil's talk at the summit in Boston, which is on YouTube now, goes into this in a bit more detail. But it's really nice to be able to do this and move things out there. Also events. So talks like this, bite-sized seminars, training. So Geraldine Van Oera, who was at the Broad at the GATK, developing and helping out a lot with the GATK best practices, has now joined Secura as lead developer advocate. Phil has moved a bit more into sort of open source platform type of role within Secura, and so she's now going to be driving a lot of these efforts here to start engaging with community and seeing how we could help, training, various other things that are really important. And lastly, thank you for listening, and thank you for inviting me to speak.
We've got a bit of time, so do we have any questions? Priyanka.
Okay, so moving forward in terms of, how would you say people who are coming from Nextflow to NF Core can start? What's like the first step to contributing to NF Core? How can they be part of the community in their learning cycle?
I mean, it's probably not the answer you want, but just ask lots of questions. Don't be afraid to ask questions. Everyone at some point, including me, was completely, head spinning about what's going on and how to do things. But if you ask questions and are interested, then we've got all of these community channels where you can come in, join the community Slack, join the Nextflow Slack, join various other places that are useful to you. And we've also got tons of documentation as well online that you can follow, you can get up to scratch with. If the documentation isn't right, please let us know, because the fact that you're a beginner means, I almost think that beginners write the best documentation because they have to go through that process themselves, and writing it down in a nice, easy, understandable way is priceless for me. And so if you're confused about anything, ask questions, but join the community first. Each pipeline has its own channel on the NF Core Slack, for example, and you can join there, ask questions about, how you'd like to get involved, any questions about the pipeline. There are various other channels there as well.
Yeah.
So you said there are another duplicates of pipeline. You have only one pipeline for people and city. I think it's good, and it makes you some sort of gatekeeper. You have a great responsibility. So my question is, how do you ensure that the pipeline that you unique pipeline is having the best food that can possibly have? Because there are those
So the best way, again, is just to have this conversation in Slack with the pipeline developers. Some pipelines will be outdated. A lot of, I didn't say, but a lot of people contributing to NF Core, it's complete voluntary contributions. And so they all have day jobs as well, whether they're using these pipelines for their own research or whether they're using them or contributing back because they've got some funding within the lab to contribute, or they're working as part of a wider organization like BobReg or the Tree of Life. And they need to, improve the tooling and the pipelines in some way. What I would say in that case is just ask on Slack. If you are comfortable adding that, again, this is the big advantage of having an open community, is that these pipelines are sitting there. They may not be up to date, but you can always contribute as well. Push issues, ask questions, create pull requests, and the pipeline improves over time. So in general, what gets added is a community decision. And in order to make that decision, you need to have a conversation somewhere, and Slack is probably the best place to do it. If we can get contributions, then that's a massive bonus back out there.
Brilliant. We're running out of time, so we're going to move on to the next talk now. Thank you very much, Herschel. Where's your pipeline? Which one's you?
Thank you.
There you go. So now we've got one button. So now we've got Matthew and Damon from the Tree of Life to talk through resource optimization and CO2 awareness across Tree of Life pipeline.
Hi everyone. So it's going to be two speakers. We're going to share the slides. How it's going to be smooth enough. So right. So we are the Tree of Life program, the Sanger. One of the main. Can I move? Yeah. Easier. Right. Thanks. So we want to make 6 scientific programs, and the goal is to make reference genome assemblies for every species on Earth. So we're starting with everything in the UK and then growing. We are many teams involved in that. We're about more than 60 people, and it's about making standard processes as well. So we've already done more than 1,200 assemblies. And those are currently new species for which we didn't have any assembly so far. And so far we're doubling year on year. So next year, by the end of next year, we'll have done another 1,000 or 2,000 more. No? Okay. And then building on what Arshad just said, we're following the methodologies, the standards provided by the NF Core tools. Because they're already there, they're very good standards. They make our things portable, like you said, they can just run on the cloud. As long as you follow standards, it just works. So it's really great. And we've got all our documentation now laid out in a standard way on the pipeline website. That's a collection of close to a dozen pipelines. So here I've selected the main topics. Some of them have got posters out there if you want to talk to the authors. So it's about genome assembly, decontaminating the assembly, that's removing sequences from foreign organisms, helping the curation teams to make the best genome ready out of data, and then standard genome analysis for everything. So, Damon.
Hello. So I'm one of the 3 developers on TreeVal. We're currently running up to our 1.1 release called Ancient Aurora. And we sit between the decontamination of genome assemblies and the manual curation of those assemblies to effectively turn them from scaffold-based to chromosome-level assemblies. And to do this, we generate a lot of data, quite frankly. We make 46 different files, including their indexes, across a variety of formats in 13 major sub-workflows, 5 minor sub-workflows where they're essentially a sub-workflow nested inside another sub-workflow, and well over 100 modules at this point. For those of you that have seen the poster outside, I hope you can attest it's a pretty big pipeline. And we shovel all of this into JBrowse 2 that you can see up there. So each of those is a different data type that a curator can use to improve the contiguity of a genomic assembly. And these are some more. So we've got, it also generates high-C maps from Juicer for high glass and for pretext, which is a modifiable high-C map. So far, TreeVal has been built kind of halfway. Well, it started about a year and a bit ago now. Previously, we used something called GVal, which kind of died unceremoniously, in all honesty. But since then, we've been working on TreeVal, its successor for the Tree of Life. And we have done 70% of the assembly so far. Eventually, we will do everything. So that will be the 70,000 species that we estimate are in the UK and Ireland. And for each run, we use, for each species across the entire toll pipelines, we use about 8,000 to 10,000 CPU hours. For everything, that will be 630 million CPU hours over the next 10 years. This excludes the monsters like mistletoe, which took literal months to shovel through the pipeline. Obviously, this is a hell of a lot. So any sort of optimization we can do to bring this down will help out us, it will help out our infrastructure teams, and it will help out other users of the farm. And the issue is that we've come across is that Cicera platforms, their optimizer on there only does one pipeline run at a time. So, do you want to do this function? Yeah. Which I will come to more in a minute. The suspense.
The NF core modules we're using define our resources as low, medium, high, and have bins of 6 GB of memory each. Actual usage is usually not exactly a multiple of 6 GB, so you're bound to have a bit of waste there. But it's not just that. Actual usage is usually dependent on the input of your module or your software. They can be quantitative values like size, length, number of sequences, but also qualitative ones like just a clade name or things like that. And having unoptimized pipelines costs us a lot. If we underestimate resources, then the process will crash, and we have to be restarted, maybe multiple times. If we overestimate, and that's a lot of resources that we've asked, that we're paying for, that actually we're not using. So we really care about using our HPC and the resources efficiently. That's why we're doing all these efforts.
So to combat this, we decided we needed to add some context to the execution logs. So what I've done is wrote a very simple, groovy module, which we shoved into the pipeline that collects some very basic statistics on the input of the pipeline. Obviously, by itself, this means practically nothing, but we have run this across 350 runs of the pipeline so far. And we push this through the summary stats script and generate some graphs for some actionable insights. Currently, we produce about 40 of these with plans for many, many more. But ultimately, I need to be able to drill this down to a per module, per process, add the max total resources against the peaks and requests. We also use the CO2 footprint module plugin that was mentioned earlier to kind of, we're DTOL, we're Darwin Tree of Life. The whole point of this project is to empower conservation. We can't really empower conservation if we're wasting resources by the bucketful. And both me and Matthew essentially started optimizing these pipelines in our own ways and have kind of converged upon making a ton of graphs, which is quite nice. So this quite horrific looking graph, actually, this is an output of the script so far. So on the left, we have all the unique clade counts so far, the types of run, because TreeVal is run both in a full and rapid mode, depending on the data available, and the projects they belong to. So this is of one of the sub workflows in TreeVal that takes Hi-C data and maps it against the genome. The blue dots are it's the peak memory as a percentage against that requested. And the box plot is the average used memory as a percentage of that requested. So as you can see, that looks pretty horrific. We're using on average, maybe 30% of the memory we're actually requesting. So this slows everyone down. This means we can't get as much TreeVal runs done at the same time as we want to, makes other people on the farm wait for their pipelines to run. It's just not good all around. Some more examples. These are just some other examples of graphs that we generate. So Hi-C against duration, to try and see whether the total amount of Hi-C impacts it. It doesn't much. That gets pretty flat pretty quick. And that's just because it's parallelized. So pretty much every time Hi-C data is getting split into at least 6 chunks, run at the same time. It's pretty happy. We're also doing some categorization of these runs to see if there's anything that we can drill down to. Insects take X amount of resource, or as you can see, fungi are on average much less than the clades around them. This is an actual usable graph. So this is the amount of Hi-C against the amount of memory, the peak memory for that sub workflow. So using this, we'll be able to plug the trend line into the config, and we'll get it to start dynamically requesting X amount of memory per gig of Hi-C data, which is quite cool. And obviously, we've categorized this down to the clade level, and there are some slight differences. Now, the ones that will make IT hate us. So this is a graph of the percentage of CPUs requested and actually used. So this is the request CPU numbers as a percentage more than are actually used. So 100% is where you would want this to sit at, or probably a bit above that for some wiggle room. But on average, we're using about 160% more CPU than we actually need. And on the memory side, it's even worse. We're using, on average, get rid of the outliers because they're probably from runs where I've massively increased it for a specific reason, which I've forgotten. But get rid of those and we're probably looking at 750% more memory than we actually need for on average. Pretty horrific. So far, what we've done to combat this is to add a process tiny. There are umpteen jobs in the pipeline which are just simple bash scripts, so awk or find or very small stuff. These need next to nothing in order to run. We've got 100 meg there, but that's very much as you should never need more than this. Gene alignment has been modified to be slightly more aggressive than other tasks. We multiply the task attempt by 1.5, so it has a slight curve on its requirements. And many more will come as we collect more information from the summary stats. This is something that I've still got in fairly early development, really. But the important thing to note here is that we only found this because we were looking. There's obviously, I should imagine, all of us write Nextflow pipelines. How many of us are actually thinking about optimization of resources? How many further still are then collecting statistics on their resource usage? And then how many further are doing what we're doing and trying to generate something to improve that?
Yeah, there are solutions, don't get too depressed. So Nextflow is highly configurable. So this is an example here where we can say, okay, so my default is going to be 30 minutes for each process, and then 1 hour for these ones, 2 hours for those ones, and so forth. So Nextflow allows targeting specific processes and giving them tuned resources, either in a constant manner like this, or you can make it depend on the input size. So this is an example where I say, okay, for smaller genomes here, I want 4 gigs of RAM, next category, 8 gigs, and so forth. We can make it a nice correlation against the input sometimes. So here, I want 50 megabytes of RAM for every 500 megs of sequence. This one, I increase the CPU, depending on the genome size and memory as well. So we can target specifically. I know something like this here, it means that, and I've checked that by looking at the source code, I can guarantee the usage of my tool is going to be within 50 megabytes of what I requested to the HPC. So that's very close to the optimum. In this case here, it's more about thinking about after multiple attempts, or thinking about the efficiency in long term of your processes. Multi-threaded jobs usually are becoming less optimal as you increase the number of threads. So you don't want to increase in linear manner, and instead I want to increase in a logarithm manner. That's what I'm trying to do here with BWMem. But then there could be a link with memory. Samtool sort. Where is it? Yeah, needs 768 megs of RAM per thread. So wherever I define here the number of CPUs, I need to add that here as memory as well. You need to think about how those things are linked together. But what we realize as well is that it's not just about number of memories the amount of resources you request for each process. It's about how the sub-workflow, the pipeline works. The standard NF core way is good. You've got one module doing one task, and then you run, so it runs one, makes an output file. That's then used for the second step, it produces an output file, and so forth. This is a very standard Samtools sub-workflow that's advertised on the Samtools documentation page. That bash pipeline cannot be shared through NF core, so it has to live as a local module within a pipeline. So it's something that you need to be aware of, that's an issue, and then you need to do an extra step to go and implement it in your pipeline. But it makes massive differences, because this is 5 times faster than having data separate jobs. And then you save 3 independent BAM files. So you save a lot of space as well. I've got another example here that's about parallelizing your BWMem alignment across multiple machines. So there's a trade-off between modularity and performance. So we start with Nextflow for modularity, and then as we optimize pipelines, probably we're losing a bit of that, but there are other benefits, obviously. So here, this is the outcome on 2 of our pipelines to do make statistics and figures for our publications called genome nodes and read mapping. You can see a total amount of memory requested across my test data set is divided by about 4. So we're using 4 times less memory as initially. Number of CPU as reduces as well. So it's really the massive gains to be made. And like Damon said, if you overestimate your resources, it makes the whole HPC, the whole cluster go slower because you take resources away from other users. But I still have some questions there, and I hope we can discuss that. So for instance, so typically what we do is that when a process crashes, it will then restart with more memory, more CPU. But I would like to have to fine-tune that. If it's killed because it's using too much memory, then yes, I want to increase the memory, but don't necessarily want to increase the CPU as well. Or if it's killed because it increases the runtime limit, then I want to give it more runtime or more CPU, but not necessarily more RAM. Some tools may exceed because of internal error, like a segmentation fault. In that case, maybe I don't want to increase resources. It depends. I would like to have more flexibility there. What we're aiming so far is about finding resources so that every process works at the first attempt. So that would still be overestimating resources in some cases. Maybe it would be fine to actually decrease that amount and allow a few processes to restart. It's about, again, another trade-off there. I don't know how we didn't do a raise your hand, so I don't know exactly how many of you are optimizing your pipelines, but we've got some outputs there. Can we share that with the community with NFCore? Is there space somewhere to deposit that? And then finally, we're doing this after having run the pipeline in an unoptimal, very suboptimal manner for several months now. Can we do that without running it at all? Is there a magic AI tool that can take a source code and tell me, how much memory it's going to use? I don't know. But I would like to avoid wasting resources in order to the whole thing.
I will try to be very quick, because I'm very aware that I've spoken far too much already. So as we've already said, CO2 footprint plugin, it's going to be very important for us. We need to be more sustainable anyway, because we're killing the planet, and it kind of gets the antithesis of DTOL. I need to generalize the script that I have so far. So at the minute, the script I have is very geared towards TreeVal. However, TreeVal it's a big pipeline that, anyway. A lot of graphs. We need more graphs. Efficiency per process. We need to be able to search through our data sets via date, pipeline versions, most pollutant, and the clades. And what should we do next? Should I turn this into a plugin so that it collects data on declared channels, and then it stores that data somewhere in a way that is easily that we can easily stick together and create massive data frames from? And is it possible to eventually add custom scripts pages to platforms? So then platforms is already there aggregating everything. So if we could do this, then we could have my script hook into it. Bob's your uncle. And finally, thank you.
So we're not going to have time for any questions. We're going to run away quickly onto the next presentation. Which I will now look for. Damon, do you know where here we go. Not named. Here we go. So next, it's all right. So next we have Mattis, who's going to give us from HumGen here, Sanger, who's going to give us a talk on, here we go, on
YASC, which I don't know what that stands for, but I'm sure he will. And hide voting meeting rules. There you go.
Thank you. Yeah, it's actually a very good question. What does YASC stand for? So it stands for yet another single cell pipeline. So essentially, we were utilizing this pipeline to analyze a large-scale single-cell project called Cardinal, where we basically sequence UK Biobank and ELJH individuals, 6,500 in total. How do I move? Okay. So this is me. I'm principal bioinformatician in human genetics informatics team. Work for Vivek here. And one of the largest projects for single cell was Saronzo team's project, which is UK Biobank's sequencing of their participants with the single cell technologies. Hence, we wanted to develop a large scale single cell pipeline to be able to approach 12 million cells essentially and make sure that we can QC everything and get stuff done. And Nextflow is perfect for that. So just a very high level overview of Cardinal project. So it's, like I was mentioning, it's sequencing of UK Biobank and Genes and Health cohorts. So essentially, they're collecting data from multiple different places within the UK, shipping everything to Sanger, and then sequencing things. Saronzo team developed a lab pipeline that was capable in organizing blood in a very well-mannered approach and record all the metadata within Sanger and capture it in databases. And then we came in to essentially develop a large-scale single-cell analytics pipeline, which is capable in QCing the data, ensuring that everything is going to the right place, to the right individuals. So, like I was mentioning just in the previous slide, UK Biobank and Genes and Health. UK Biobank is essentially a very large resource of individuals with the metadata and lots of associated lifestyle questionnaires. And it comes mainly from wide British background. Genes and Health is South Asian communities in East London. So it's 1,500 individuals that we're sequencing. But again, there's a vast amount of information on the metadata for it. And what we developed is this yet another single cell pipeline that is mainly developed for Project Cardinal, but it's applicable for many other projects currently in Sanger. One of the upcoming ones is Project Jaguar, where essentially the idea is to sequence around 1,000 individuals around South America. So we're adjusting the pipeline to account for that. And the benefits of the YASC pipeline is that it's actually very nicely integrates within the existing pipelines in NF-core, which is single-cell RNA-seq pipeline, which is capable in aligning the PAM files and everything like that. So we're biting on top of that and basically taking the cell ranger outputs from the single-cell RNA-seq pipeline in NF-core and performing the QC metrics that are necessary for our particular project. I tested it on PBS, LARM, and LSF just because I use this pipeline in Cambridge, Leicester, and Sanger, and it works perfectly fine. And the benefits of that is basically NF-core's template that I utilized because I can easily, not even thinking about configs or anything like that, pick up this pipeline and run it in different cluster, and it works very well. So what we are basically trying to do, this is a little analogy, but essentially every single individual in the blood has multiple different cell types. So we can imagine those as an ingredient in cooking. And then what you do is you basically multiplex multiple individuals together up to 12 in Cardinal. So we're basically cooking a lot of people in the same oven. So what you're trying to do later on is basically take these individuals apart and make sure that every single cell is belonging to the correct individual and there's no contamination within each of the particular parts. Pipeline is quite massive. I'm gonna touch on multiple different bits, but essentially you can easily, in Sanger, you can run it with just module load the pipeline and then provide your input file and YASC run. So that's the benefits of providing the pipeline to other individuals within Sanger. It's an NF-core template used. So if you actually clone the pipeline, you can just run it, Nextflow run and input files. So I need to improve some documentation on how to run some of the multiple aspects of it, but it does loads of different things that I'll briefly touch on. In Sanger, we have, like I mentioned, captured all this complex information in multiple different databases. So we have another pipeline that I'm not going to talk about, but essentially produces the correct inputs for YASK for us, capturing all the different information about like which sequencing center it came from, how much data is there. And then we run the YASK pipeline, which QCs everything. And then we have an internal website where we basically track how well the performance on each of the runs has gone. So like I was mentioning, pipeline is modularized, so you can actually bite in different bits of it, because sometimes people don't need the entire pipeline, they just want to run bits of it. So within Sanger, again, we have modularized these little bits of it. Some of them is not fully finished, but like if you wanted to just remove the ambient RNA, which is basically stuff like if cell bursts and then some RNA leaks within the droplet that you're incorporating and you want to remove it, then you can actually run it easily just by entry points within Nextflow pipeline, or we have wrapped it around it with the modules and you just say YASK cell bender an input file, and then it's just going to run that particular bit of the pipeline. So yeah, like I was mentioning, it's basically removing this stuff that leaked in other individuals' droplet and ensuring that everything's removed that's not supposed to be there. And then you can also run deconvolution bits. So this is basically taking the donors apart. So when we multiplex 12 different individuals together, we want to make sure that donor 1 is only containing donor 1 cells, donor 2 is containing only donor 2 cells. So you can actually run this little bit of that as well. So you can just say Nextflow run and deconvolute. And when you do the deconvolutions, I'm not sure why all the images disappear, but essentially you also perform the gene tapping on each individual cells. So you can ensure that the cell belongs to the right donor, because you can start to look at whether the genotype is concordant or discordant with the donor. So once you deconvolute the individuals, every single donor has its own cells. And then this is another bit when you start to assign the cell types. So like I was mentioning, every single blood contains multiple different cell types. So you can start to take them apart using azimuth and cell typist. You can assign what cell type is which. So now if there was images, you would see that these are different cells. So every single donor has the right cell assigned to the right donor. And then we're performing QC metrics down the line, which is basically removing the cells that has a bad QC or is not sequenced well or is not aligning with the correct donor well. So we're ensuring that that's happening. Just some images that is produced by the pipeline. So essentially removing the blue bits and leaving the red bits. And the last bit is integration. So as the time goes and you sequence a lot over 2 years, then you start to see the batch effects. So you want to start to integrate the data, normalize it based on the batches. So that is the bit in the pipeline over here. And you can again run it independently of the pipeline. These are some of the bits. So it assesses how well it clusters and integrates within the pipeline. And like I was mentioning, you can also utilize this for multiple different other projects. We reprocessed a large-scale single-cell project 1K that produced some interesting results, and the pipeline can be utilized anywhere else. And we also working with SCverse, and they were quite interested. So that's another community within Sanger that is basically for single-cell analytics tools and things like that. And yeah, that's it. So we managed to process 11 million cells with this pipeline, and there's loads of efforts coming from multiple different aspects to get this thing done. Thank you.
Thank you very much, Mattis. Again, we're now running slightly behind, so I'm going to move straight on to Lithika, who is going to talk about variant ensemble predictors with Nextflow.
Hi again. I'm going to formally introduce myself. I'm Likita. I work as a bioinformatics developer, and we provide genome annotations as part of the Ensemble project. Yeah. So today I'm going to talk about our work on speeding up variant annotation in the Ensemble variant effect predictor using Nextflow. So analyzing genomic variants is essential in terms of patient care, surveillance, and treatment outcomes. And the Ensemble variant effect predictor is a powerful tool set for analysis, annotation, and prioritization of genomic variants. It predicts variant effects on the gene using several algorithms and reference data. It can get tricky when the input file is large. And imagine a scenario when you've been running the tool for about a week and it errors out on the last day. That's not ideal. So we've come up with wrapping the variant effect predictor using Nextflow. Nextflow Ensemble Web Pipeline was released in April 2022. It currently supports multiple job schedulers, including LSF and Slurm. And we made use of containerized images to remove the overhead of setup and installation issues. The parallelization was initially supported at a chromosome level, but because it was not a balanced parameter, we shifted to something called a bin size. So depending on the bin size, the file is split accordingly. And it turned out to be scalable and faster compared to our traditional pipelines. To give you an example, about 7 million variants took about 48 hours. It was previously taking a week for us to annotate 7 million variants. And Nextflow's resume functionality allows continuation of the workflow from the last successful step, as you might know. So that's useful. To give you an idea about the pipeline itself, so the pipeline takes an input variant file, and it performs some processing, pre-processing, and data validation checks. And depending on the user parameter or a default given bin size, it splits the input file into multiple sub inputs or sub files, and then runs Ensemble Web on those multiple sub files, and finally merges it into one single output file. So we initially, and this is one of my first projects, so it is kind of special in that way. And we initially started off with handling the use case with a single input and a single configuration file. So the Ensemble Web can take a configuration file. But as we continue developing, we realized that we have to handle many more use cases. So in terms of annotating pan-genome assemblies, we wanted multiple configuration files having different assembly information in those configuration files, but starting with a single reference data. We also wanted to do some benchmarking, and something like benchmarking required testing it on multiple input files and so on. We are still in progress regarding supporting both multiple input and multiple configuration files. This was a bit tricky, so we are still working on that. Some of the optimizations and enhancements that happened since we started were, first of all, like I mentioned, we converted taking chromosome as the input, and we shifted to splitting by bin size. And often these pipelines generate a lot of intermediate files by the tool. While there is a way to clear up the work folder post the run, there was nothing that I was aware of to clear out the space while the process was happening. So we used AfterScript to remove those files as the process was happening. And for the merging step, we initially used BCF tools, but it turned out that the Linux, there was a strategy to sort within Linux sort, which was better in terms of speed. So we improved the merging strategy with that. And in terms of user experience, like I already mentioned, we improved the use cases that we were going to handle. And the pipeline currently supports non-compressed and non-indexed files as well. We now support relative paths in both the input and config files. and we declared the workflow as a sub-workflow, so it can be invoked by other pipelines. You must have enjoyed the talk by my colleague Nuno, which was about protein function prediction. So after running the data using the protein function prediction, it can be run with Ensemble Nextflow Web to annotate on the variants. So that's one way of using this pipeline in the downstream pipelines. Coming to our future work, we want to improve our unit tests and utilize NF tests for that. We would like to extend support to cloud and Kubernetes deployments. While this is not in our immediate plans, we would like to look into it. And we want a better managing and reporting tool in order to manage the Nextflow pipelines. So we would like to integrate it with Nextflow Tower. And that's all I had. Thank you, everyone who has supported me, especially the funders as well. Thank you.
We have time for a quick question, so everyone has a question. Anything on chat? Okay, cool. We'll move on then. Nicely timed. Thank you, Lithika. So the final presentation of this session is Simon Pearce from Cancer Research UK, who is going to talk to us about his work with cancer methylation and detecting and subtyping cancers.
Okay, and my slides are already slightly worryingly, the text is dropping off the bottom, so we'll see how we go. So yes, this is a more biology focused with bits of Nextflow here and there. So hopefully you'll get both the biology story, but also the Nextflow bit. Hoping I've balanced it right. So yes, I'm going to talk about circulating cell-free DNA methylation and using that to detect and subtype cancer. I'm part of the Cancer Research UK Cancer Biomarker Centre, which is connected to the Cancer Research UK Manchester Institute and is all part of the University of Manchester. Okay, so circulating free DNA is bits of DNA that's just floating around in the plasma in all of you. So everybody has some, but this is increased in patients who have cancer. And some of that increase comes from the cancer cells. And so you can study the cfDNA, the circulating cell-free DNA, to learn something about the cancer. And these methylation changes often occur early in the cancer development, and the amount of tumor fraction in a patient, the amount of cfDNA that comes from the tumor can be very variable. And one of the things about this methylation changes is they typically occur early on in cancer development, and they can be quite consistent between multiple different patients with the same cancer type can have the same changes, whereas mutations, they might have very different driving mutations. So within the Cancer Biomarker Centre, we've developed an optimized methyl capture sequencing approach to profile the whole genome-wide cfDNA methylation patterns. So we take the DNA, we add on at the very first step. So this cfDNA that you can extract from plasma can be as low as 1 nanogram in a blood tube. So there's very low amounts of DNA. So what we have to do is we add on a sample barcode so we can pull the samples as the very first step. And then we can perform, like we have sufficient material to perform a methylation enrichment using a methyl binding domain protein in order to pull down the regions of the genome, the fragments of the DNA that are methylated. And then we can profile that. And this is a bit like ATAC-seq, ChIP-seq. We end up with reads in certain regions of the genome. And so this looks like this. So you can see here a tumor sample at the top, and then the patient's adjacent bit of normal lung tissue next to it, which has peaks in different regions. And so we take that, we take 300 base pair windows is what we typically work with. And then you can see there are differentially methylated regions between conditions. So you can see there's this region here that's methylated in the tumor, but not in the normal lung. And then you can look in patients. So this is a different patient to who the tumor and normal lung is coming from. But you can see there is consistency between that tumor sample from a lung tumor and then a patient, the cfDNA coming from a patient. And then it's absent in this non-cancer control sample here in cfDNA. So in terms of what we do with that data, so I started this work 4 years ago, and it's grown and grown. Initially, I had a DSL1 pipeline where it was just everything stuck together. I now have 2 separate pipelines using the NF-core tools, which are extremely useful. Like, in order to, the tools is the unsung hero, I think, to some extent, of NF-core. So I have one DSL pipeline to do this demultiplexing. So because our barcodes go inside the FASTQ files, so they become the start of the FASTQ files, I end up, I have to demultiplex them to say which sample they came from originally. And because I'm impatient, the qualities talked about early, I want to parallelize that. So I actually split up the FASTQ files into chunks. And in order to run UMI tools extract, which is quite slow and single-threaded, it isn't parallelized. So I chunk it up in order to put through the same sample through these tools, and then I stick them back together and then demultiplex. Then after I've run that demultiplexing, I then have normal FASTQ files, some of which are methylation enriched. So those have the peaks. Some of them are just sort of shallow whole genome sequencing. And then I run them through a pipeline that we built in-house to analyze this kind of data. And I'm going to highlight, because I really like talking about it, the tool NGS Checkmate. I had a poster outside. So it's a tool to check that samples match when you expect them to or don't, or tells you whether they do or not. Okay, so I want to then use this for cancer of unknown primary. So these are patients with cancer of unknown primary. They have been diagnosed as having cancer, but despite a lengthy workup time, diagnostic workup, they can't figure out what original cancer type it was. So this might be because the primary site is too small to be seen through imaging in a really hard and awkward place to image, or it might have been cleared by the immune system and no longer there. Or it might just, because it's cancer and therefore has mutations, it might not be showing the typical markers, the typical things that make it look like a certain cancer. And so these patients just get standard care chemotherapy and they don't get access to newer treatments and targeted therapies because they don't have a diagnosis. So how can we improve that diagnosis? And particularly as tissue biopsies are often very small, they're taken from the metastatic biopsy, metastatic sites, they might be quite small and necrotic, and they're used up in the diagnosis process. So we want to use cfDNA for that. So can we predict cfDNA, cancer type from cfDNA using this cfDNA methylation signal? And these methylation patterns vary across tumor types, both due to the tumorigenesis process, but also there's quite a lot of background tissue signal. So this DNA methylation is a way that your different cells with the same DNA express different proteins. So we need lots of data for this. So where do we get that from? We get that from TCGA. So TCGA, the Cancer Genome Atlas, was a massive project that sequenced about 9,000 tumor samples with methylation arrays. So that's methylation arrays. It's an older technology. It measures at fixed locations on the genome and calculates what are called beta values, measures the methylation between 0 and 1. And it's been shown, so people have taken the TCGA data and shown, oh, look, you can see tissue-specific patterns, as you might expect. So I need to convert them. I have this T7-MBD-seq profiles, and I want to do something with arrays. And so I have there's a software tool a package called QSEA that lets me go from reads to beta values. And so I do that in reverse. I go from reads to beta values to give myself a lookup table, and then I take the beta values I get from the published data and convert back to counts to make pseudo profiles for each array. And so when I do that, I take the data from TCGA, I convert it, and it's still, I get a nice UMAP sample separating by the tumor types, as you might expect. And that structure remains if I take the total array and downsample to just the most strongly different regions between each cancer type I'm interested in. And here I've got 29 different cancer types, like I've regrouped some. And so I've now got 22,300 base pair windows across the genome that I'm interested in. And I want to build a classifier. So I have these arrays that I've taken and I've converted them to be equivalent, mostly from TCGA, but a few from Gene Expression Omnibus. And I have some non-cancer control samples. That's cfDNA from volunteers who don't have cancer. So that's the sort of you and I typical background signal. So I then do, because I'm not interested in applying this to tumor samples, I'm interested in applying this to cfDNA samples from patients, which might have 1% of their cfDNA might come from the tumor. So I do a data augmentation strategy, which is mixing, and I mix the array data into cfDNA at small amounts. So 1%, 2%, 5% to make these synthetic mixtures. And I do this lots and lots of times. I also mix non-cancer samples with other non-cancer samples, again, to increase my training population. And I end up with about 280,000 mixtures for this particular classifier. And then I subset those mixtures because like I've taken my original 2 sets of data and I've expanded them into a massive data set, which comes from all of those. So I might want to make sure I'm not overfitting. So I take subsets of that and train lots of individual classifiers. I train 100 of these sub classifiers and then average over those. And that gives me an ensemble classifier that has 30 different tumor types, and I make predictions for a sample. All right, the classifier thinks that it's a lung cancer or a breast cancer. And the performance looks good when I apply it on sort of held out mixtures, but how does it perform on actual cfDNA samples? So I've not trained it on actual tumor samples, I've just trained it on these mixtures. And so when I apply it to actual samples, so I have 143 samples from known cancer patients of known type. So they're on the left here. So you can see the predictions that are made in the middle. I guess I should mention the classifier is called Cupid or CupID. So I have most of the predictions that I make, if I make a prediction, are correct. And then I don't make predictions on additional non-cancer controls. So I get really nice sensitivity and specificity in using cfDNA from blood samples to determine cancer type. So applying to actual patients with cancer unknown primary, I make 15 different predictions out of 41 patients. And mostly these are quite typical things you might expect in patients with cancer unknown primary. And they're clinically consistent. So these patients have a lot of diagnostic workup, and they may well get a differential diagnosis. So they're suspected to have this or this, like breast cancer or a gynecological cancer, but they're not sure which. And our classifier is generally consistent with these predictions. We will predict one of those two. And in the patients that actually have a resolved diagnosis, so 15 of our patients get a later diagnosis. Of those, we predict the same thing in 10 of them, and 2 of them we predict something different, and 3 of them we don't make a prediction. But these take the median time to these actual diagnosis for these resolved patients is 7 months. So it's a long time of suboptimal chemotherapy and diagnostic uncertainty. So that's my actual application. But then moving back to Nextflow again, I have recently, since sort of that work, the building with that was all a set of shell scripts and R scripts. So I now have, using the NF Core template and tools, a pipeline entirely custom, but using the NF Core framework and structure to do the same thing. So I can build classifiers using raw IDAT files, so that's the methylation array data, if they've been deposited in the libraries, or I can use processed values of beta tables if they haven't deposited the raw data, or I can include our samples we've sequenced, and combine them together in a Nextflow way. And I can now build classifiers for different questions much easier. So to summarize, I have we have this method that can profile methylated regions across the whole genome, suitable for cfDNA. I've got these three different Nextflow pipelines that I've used, all based on the NF Core tooling, just to emphasize how useful that is, just being able to grab the modules that you need and then build extra ones for the custom bits. And so I can convert arrays to be compatible and then build classifiers, and we can accurately classify tissue of origin for these cancer samples from plasma. And this could help in patients with cancer of unknown primary. I have my other half of my acknowledgements. Okay, there should be more on the right-hand side. I don't know where that's disappeared to. Okay, so I need to thank particularly Alicia Conway, whose PhD she's a clinician, but she did a PhD and she did the lab samples. Alex for driving the wet lab development and optimization, and Stephen, Paddy, and Kate in particular from the bioinformatics side. And on the other half of the side, the patients who donated, and their families who've consented, the scientific computing at CRUK, Manchester Institute, and the clinical team for collecting the samples. I think that's everybody who should be on the right-hand side. Thank you.
Right.
And NF Core.
Yes. Do you have any questions? No, great. Then we can move on to a refreshment break where I believe there are sweet things that have been promised for you all. So please, we'll be back here at 3 p.m. for the panel discussion. Apparently not. I looked. Thank you very much, Oshan.
I stayed on time.
Really?
I stayed on time.
How do I? Yeah, I'll mute it now.